Okay, I think we're going to get started. My name is Nevin Krogan. It's my pleasure to welcome you to the QCRG COVID-19 Research Symposium, which is being sponsored by the Quantitative Biosciences Institute at UCSF. As you can see, we have an exciting lineup of speakers for you today from all over the world, talking about different aspects of COVID-19 research. I would like to start by thanking our sponsors for this event, Brooker, Synthago, Thermo Fisher Scientific, and this event's being coordinated with the Host Pathogen Mapping Initiative, which is based out of UCSF and UC Berkeley. And I'd just like to start by telling you a little bit about the Quantitative Biosciences Institute. It comprises over 100 different labs here at UCSF, and I would argue it's really focused on collaboration. It's focused on bringing different scientists together. We're heavily involved in developing and using quantitative approaches to study in a disease agnostic way many different disease areas and the underlying biology behind those different disease areas, both experimental and computational. We're heavily motivated by investing in the youth. We have a number of programs in that regard. We partner with different entities on campus to provide grants that bring different scientists together. We have a strong media outreach presence, a seminar series, a symposium, and we've used our technologies in an integrated way to start 3 major cell mapping initiatives focused on 3 different disease areas: cancer cell map initiative, infectious disease, the host pathogen mapping initiative, and psychiatric and neuropsychiatric disorders in the context of the psychiatric cell map initiative. And to facilitate all of this work, we're heavily involved in international collaborations. Here's a list of some of the formal relationships that we've established over the last several years, including in Germany, in Ireland, in France, Nigeria, Poland, Israel, England. And to facilitate these international collaborations, we're heavily involved in putting on different symposia, both here and abroad. And this was our list for the symposium in 2020. I think we got through 2 of these before the shutdown was put into place. We're excited to reconvene these in the near future. But in the short term, we are now starting to do our symposia virtually. And this is our first virtual symposium, obviously focused on COVID-19. And I would argue we put together a very exciting set of speakers for this symposium from all over the world, representing 4 different continents. And we have, if you can look at our list of speakers, we have scientists focused on virology and mechanism and potential therapeutic avenues, such as Adolfo Garcia-Sastre, Daniel Swanee, and Kevon Shokat. We have some talks from different parts of the world focused on COVID-19 surveillance, including Christian Happy in Nigeria, Andrea Gamarik in Buenos Aires, Argentina, Addie Stern in Israel, and Viviana Simone in New York. It's a great pleasure to have Adave Sengupta from Gilead, who will be talking about the work getting remdesivir to the public in the context of these different clinical trials. We also have Melanie Ott here talking about a very interesting diagnostic approach for COVID-19 using CRISPR-Cas9. We also have representation from the Pasteur Institute, Christiana Gurka, talking about the efforts coming up with a COVID-19 vaccine. And finally, we have an exciting talk from Elizabeth Fisher from Rocky Mountain Labs in Montana. She'll be talking about her efforts using microscopy, electron microscopy, to study coronavirus and other infectious agents as well. So there's 2 sessions to this symposium. I'm going to be chairing the first session. Kevon Shokat is going to be chairing the second session. There's a break in between both sessions. It's a half-hour break. And there's a 5-minute talk from one of our sponsors, Synthago. It's a flash talk. And they're going to be discussing how they're using CRISPR genome engineering to help all of us study COVID-19. So I'd encourage you to listen to Synthago's 5-minute flash talk at the beginning of the break, which will be roughly around 9:45 PST. And probably one of the most important slides here is to give credit to the organizers of this event. Is that Jacqueline Fabius, the Chief Operating Officer of QBI, and then the QBI media team, Gina, Alexa, and Carolina. Thanks to this crew for putting on this event. And I'm going to kick off the symposium by telling you a little bit about the recently formed QBI Coronavirus Research Group, the QCRG, which obviously came from the Quantitative Biosciences Institute. And it initially encompassed 22 different laboratories at QBI. And since then, it has swelled to over 40 different groups, essentially encompassing hundreds of different scientists here at UCSF. And these are scientists focused on systems biology, chemical biology, structural biology, virology, computer science, just to name a few disciplines. And it's been very exciting to me to see all these different scientists come together with their different approaches in an integrated way to try to come up with solutions for this pandemic that we're dealing with right now. And this picture here with a number of us, I think it was the last picture we all took together at the beginning of March before the shutdown was put in place. So this is the last time a bunch of us were together, and we're hoping be together soon after the dust settles on COVID-19. So we've actually split the QCRG into 12 different subgroups listed here. Some of them are technologically oriented, such as modeling and mechanism, which is led by Tanya Kortemi and Andre Shally. Others are focused on biology, such as ubiquitination, led by John Gross, or epigenetics and chromatin, led by Denichi Fujimori and Alan Ashworth. I just want to talk to you a little bit about one of these subgroups, which I think is very exciting. It's the structural biology subgroup, and it's actually really just turned into a consortium because it's grown to be so big. And it's led by Klim Verba and Oren Rosenberg. And in total, it involves 11 different laboratories here at UCSF focused on structural biology. And their pictures are listed here. And we have a number of pipelines that have been created, including just systematically expressing the viral proteins one at a time in bacterial cells so that fragment screening can occur or crystallography can be used to characterize the viral proteins. We're systematically expressing all the host proteins that we found in our protein-protein interaction analysis in human cells, also for crystallography and cryo-EM. And there's also a biological arm to this set of pipelines where antibodies are being generated, mostly focused on trying to develop antibodies to inhibit the interaction between spike and ACE2. So to me, it's been a very exciting subgroup consortium here focused on the structural biological aspects of COVID-19. And these are the individuals doing all the work. It's a set of trainees that have come together in a very exciting and, in my opinion, very unprecedented way to all work together to study the underlying biology at a structural level of the SARS-CoV-2 infection. And just to show you a little bit of data from this consortium, this is an analysis that's in progress for 420 crystallography data sets collected after soaking with small molecule fragments. This is the macro domain that we're looking at of NSP3, the largest protein of the virus. It's also the protease, so it's a very druggable target. And at the top right here, I'm just showing you from the group this ultra high resolution X-ray density map. And then on the bottom is the density for a fragment bound to actually the NSP3 macro domain. So all of this information is going to be released next week collaboratively with scientists in England. So this is a nice illustration of the power of collaboration and how fast this a group that's working together can generate a very important information. This is one other project that the group has been involved in, looking at the protein-protein interaction data, looking at one of the coronavirus proteins, NSP2. We found it physically interacting with RAP1 GDS1, a protein involved in GTPase regulation, and the crew co-expressed it. This is size exclusion. You can see it forms a complex, and these are initial images of this previously uncharacterized protein-protein interaction, and hopefully there'll be a structure of this release soon. So the team has set up a systematic pipelines in order to look at all these interactions that we've identified, and we're trying to release this to the community as quickly as possible. So more to come on this front. And I'm going to just talk about one of two papers that have been connected to QCRG. The one on the left here, the SARS-CoV-2 protein-protein interaction study. The one on the right here, Daniel Swaney is going to be talking later in the symposium about the global phosphorylation landscape of SARS-CoV-2 infection. But just to say a few more words about the protein-protein interaction study. So one of the big focuses here of our effort here at QCRG is to focus on the host. There's a lot of efforts going on to try to find drugs or compounds to target viral proteins, which we're doing, but we're also looking at the host. We want to understand how the virus comes in and hijacks and rewires the host so that we can come up with hopefully novel therapeutic strategies to interrupt those important connections that the virus needs to infect our cells. And there's advantages in targeting the host. You don't have to worry so much about resistance because we obviously don't mutate as fast as viruses. We and others have found that different viruses target similar host machinery during the course of infection. So if you could come up with a strategy for COVID-19, a host-directed strategy, hopefully it could be applicable for COVID-22 or COVID-24 or other viruses that can come along. There's obviously disadvantages to targeting the host, and that's toxicity. But this is why a lot of our effort here is focused on trying to find FDA-approved drugs or compounds in clinical trials that have past toxicity issues. So we're looking at drugs and compounds that hopefully wouldn't have these toxicity issues in our drug repurposing efforts. So there's about 30 genes in the virus compared to over 20,000 genes and proteins in us. So the virus needs our genes and our proteins in order to live and replicate and infect our cells. And the goal here is to try to systematically identify all the proteins that the virus needs to infect our cells. So we've been carrying out these large-scale protein-protein interaction studies, affinity tagging and purifying each one of the viral proteins and looking by mass spec all the co-purifying human proteins. And this is a project that we did. This was led by Dave Gordon here in my group in collaboration with many others. We identified over 300 proteins doing this, and this allowed us to narrow in on 69 drugs and compounds that we thought would bind and inhibit at least one of these particular human proteins that the virus needs to infect our cells. So we cloned out all of these genes one at a time, and we tweeted out that we had this set of constructs, and we said we would send these constructs out to whoever would want them, no strings attached, no MTAs, please feel free to distribute as you wish. And we had a number of groups take us up on that offer. And in total, over the last several weeks, we've sent this set of plasmids to over 350 labs in 41 different countries. And I would say that these plasmids split around the world much faster than the virus did. So we're very happy that we're able to help scientists around the world expedite research on COVID-19. So as a community, we could come up with solutions to this present pandemic. So here's a look at the protein-protein interaction map that we generated. This is one version of it, where the red diamonds correspond to the SARS-CoV-2 viral proteins. The circles are the human proteins. If the circles are orange, we think it's a drug target. I just want to point out that we collaborated with a company in Hollywood called Zoic Labs. This is a company that's known for helping to make movies. They've been involved in movies like Life of Pi and Titanic. And Biz Stone, the founder of Twitter, and Ron Conway, who have been helping us along the way, got us in touch with this particular company. And they took our data and in a very interactive way, they visualized this information. And it's a very intuitive way to look at this data. You can look at the individual proteins, you can look at any drug information, you can look at any structural information attributed to any of these proteins. So if you're interested in this data, I'd encourage you to look at our paper and look at this particular website to look at this information. And we're putting more and more information in this database and this website as we speak. So one of our goals here, as I said, was to take this map and then try to identify drugs and compounds that would bind to the human proteins that we think the virus would need. And this was work spearheaded by Kayvon Shokat, who's speaking at the end of the symposium today, and Brian Schweikert. And these are two fantastic chemical biologists here at UCSF, and they identified these drugs. And we've been working with Marco Vignuzzi at the Pasteur Institute, in particular working with Veronica, as well as with Adolfo Garcia Sastre, who's speaking after me, working closely with a research associate in his group, Chris White. So we wanted to test these drugs and compounds that were predicted from the map. And just very briefly, the assays that were done in both New York and Paris, we were initially looking at Vir06 cells. These are African green monkey cells. There's now ongoing efforts looking at human cells, but initially it's just looking at Vir06 cells, growing up the cells, adding drugs 2 hours before infection. You can see that there's two different MOIs here in New York and in Paris. It's 4 times higher in Paris. Let the assay go for 2 days and then use formaldehyde to fix and stop the assay. And in New York, they're looking at microscopy and looking at a protein, an antibody, NP as the readout, and also doing TCID50. And in Paris with Marco and Veronica, they're doing qPCR, RT-PCR, looking at the viral component of the virus, or the RNA component of the virus, and also doing plaque assays to look for infection. So working closely with these two groups, we narrowed in on two classes of drugs initially that we're very excited about. One set involved in protein translation, including zotadifin. Kayvon's going to be talking about this at the end of the symposium today, as well as ternatinib 4. It's a compound synthesized by Jack Taunton here at UCSF. Both of these inhibit protein translation in our cells. And the second group of drugs and compounds that we're very excited about are ones that are known to modulate or thought to modulate these two receptors, sigma-1 and sigma-2. And this is work being spearheaded by Brian Shoykat here at UCSF. And in this particular class of drugs and compounds, we have antihistamines, antipsychotics, anti-malarial drug, there's our friend hydroxychloroquine, the female hormone progesterone, anti-anxiety medication, as well as some very exciting preclinical compounds that we're looking at right now, including PB28. And one comment I want to make about this class of drugs and compounds, if you were just look at the chemical structures of this group, there's no way you could group these all together. But if you knew what the target was of all of these, and we think we do, that's how you can group these together. And this is a great point that if you know the biology, and this is what we're trying to do, is it'd be data-driven with respect to our drug discovery and drug repurposing. If you know the biology, you're so much further off with respect to the pharmacology. You can go back and forth between the biology and the chemistry and the pharmacology as you try to find new therapeutic strategies for COVID-19. And just to talk about some future work here, work is ongoing here to transition many of these into clinical trials. There's actually been 13 clinical trials started already based on connections that were on in the protein-protein interaction map. We're trying to start several more. I personally think a combinatorial approach is going to be very fruitful, just like the cocktail approach was for HIV. And maybe you'd have to combine a drug that targets a host protein with remdesivir, or two against two host factors, one remdesivir. So we're looking closely at which combinations could be fruitful. We're also heavily involved in genetic studies to confirm the proteins that we've identified through our proteomic work using CRISPR and using RNAi, and we're collaborating closely with Synthego on the CRISPR-based work, which again, you'll hear about at the break, at the lunch break. And we're also generating different data sets looking at different viruses, including SARS-CoV-1, MERS, and OC43. And just to show you a recent map that we generated, obviously this is the SARS-CoV-2 map that I showed previously. And just over the last couple of days, the group here, led by Dave Gordon, has created now a SARS-CoV-1 protein-protein interaction map. And very soon, hopefully in the next week, we'll have a MERS map. And it's very exciting then to be comparing across these different viruses, trying again to get at the underlying biology behind why different viruses manifest themselves in different ways when they ultimately infect us. And hopefully that'll then point to, again, novel therapeutic strategies. So this work has been done collaboratively with a number of groups, a number of academic groups, some of which I've mentioned, some of which you'll hear from today, many you won't hear about, and not just academic relationships, but industry relationships. And there's a list of companies, small companies, larger pharmaceutical companies that we've been collaborating with over the last several months. And all of this leads to just the last point I want to make with this work. And just to point out that the Quantitative Biosciences Institute has been trying for many years here to break down silos and to make connections between different scientists at different levels. And to me, it's been so exciting to see the collaborations that have been forged over the last few months. We're obviously dealing with a terrible tragedy, but a silver lining for me is to see how fast we can move when we all work together. And that's connections between different laboratories, between different institutions, and between academia and pharmaceutical companies. And I would argue that the challenge for us as scientists is to try to keep this infrastructure in place when the dust settles on COVID-19. So we're in a much better position to handle the next pandemic, the COVID-22, the COVID-24, because it is coming. And a question I have is, why aren't we working more generally like this on all diseases? Why aren't we working like this on breast cancer and Parkinson's and Alzheimer's? So I hope we can keep this type of spirit and this infrastructure in place in the future, and we can focus on a number of different disease areas going forward. So here is a picture of my group. Just a few people I want to point out who led the protein-protein interaction study. There's Dave Gordon and Gwendolyn Zhang. And I want to point out 3 individuals, one of which you'll hear about later today, who lead our mass spec efforts here at QBI, Danielle Swaney, Ruth Huttenhain, and Robin Cake. And it's just a really fantastic group to be working with here. And it's been a great pleasure to work with so many great scientists around the world over the last several months on COVID-19. So I'll stop there. And I will remind people, if anybody has questions, please put them in the question and answer box, and I can moderate getting these questions to the speakers. So that was an overview that I just wanted to give on QCRG, and I think we're going to transition now to Adolfo Garcia Sastra. If you wanted to share your screen, Adolfo. We'll jump to Christian Happy from Redeemers University. So he's leading a lot of the efforts in East and West Africa on COVID-19 surveillance. And he is a director and professor of molecular biology and genomics in the Department of Biological Sciences at Redeemers University.
Yeah, thank you, Niven. Can you hear me?
Yes, yes.
All right, thank you. Again, it's a pleasure to work with you guys on this, to present here in this symposium. I basically just want to present, talk about how we're accelerating, genomic-based surveillance for COVID response in Africa. And mark the word accelerating. The reason why I talk about accelerating is basically because, we came out of the Ebola 2016 outbreak in West Africa with the knowledge that, real-time genomic sequencing could be very valuable in order to help contain an outbreak. Early in the outbreak in 2014, we were the only facility in Africa that has the ability to sequence, and we did something that was unprecedented. For the first time during an outbreak, we were able to sequence genomes and then made the data available in real time and pioneered what we call open data access and open data sharing, which has become the norm. So within the first 3 weeks of the outbreak in Sierra Leone, for instance, we were able to sequence 99 genomes and made these genomes available for the to the scientific community to use in order to respond to the outbreak. And this really became critical in terms of helping and shaping the scientific community working in different aspects, be it vaccines, diagnostics, or therapeutics. And this, from there, we were able to work very fast, identifying problems on the ground, knowing that, PCR could take a long time, went on to work with partners to develop the first, a lateral flow rapid diagnostic test that was approved for EUA by the US FDA and then the WHO. And that the introduction of this test became a major turning point for to flatten the curve or to contain the outbreak in West Africa. Now, the lesson learned from there actually made us understand that, genomic epidemiology, could be very, very important. And not only to understand the microbial landscape, but actually to unravel and undiscover, outbreaks before they become what they are. Today we face a pandemic, but I think, if we keep building on the necessary capacity, we should be able to unravel and contain them early enough. So this, for instance, is a slide to say, for instance, where we, in our lab, for instance, we're able to use, microbial metagenomics to uncover, an outbreak of yellow fever in Nigeria. And then this was, and showing that the yellow fever strain circulating in Nigeria, in 2019, was pretty much very different compared to the ones that have been circulating in the country about over a period of 100 years. And this information was given to Nigerian health authorities. And within 24 hours, they actually declared an outbreak in the environment and effectively control and contain that outbreak. This gives you the background, how this could be very important in places like Africa, where we're always in outbreak response mode. And in this case, we're talking about a pandemic response mode. Now let's get back and then what is the situation of COVID-19 in Africa? As of today or a few hours ago, we had 275,000 cases, more than 7,000 deaths, and over 125,000 recoveries. If you look at the dashboard from the Africa CDC, you can see that the disease is all over the place, but it is not as bad as we probably would have predicted at the moment. And then what are the reasons for this? I think it's also because one of the reasons is because Africa actually shut down very quickly, but also we harnessed the tools of genomic as a way to start guiding what we call public health response. And this was a lesson learned from the Ebola outbreak in 2014, 2016, as I mentioned, and also the outbreak that was going on in DRC. But the fact that we're also responding constantly to diseases like Lassa fever, yellow fever, and then other things like cholera. So this has really put us and then made us understand that, harnessing this technology and the tool could be very important, to guide public health response. And this work that I will tell you that I'm talking about is often, or what we're talking about, the response in Africa is done in very close collaboration between, the Africa Center for Disease Control and then the National Public Health Institute. Yes, Africa does not have enough resources, to have all of the high-value gene sequencing facilities across the continent, but they leverage the poles of excellence that are existing in the continent in order to respond. So the slide that you see, for instance, is about the first case or the index case of COVID-19 in Nigeria. So this was around February 28th. Then by March 3rd, we have actually sequenced the genome and made it available. We knew where the virus was coming from. It wasn't like a transmission within the country, within Africa. But then this also made us prepared and then better. So as I'm talking to you today, we have probably not thousands, but at least with the resources available, we have about 12 countries in Africa that have contributing to genome sequencing. We have 568, whole genome sequence from these countries in Africa. But then this, basically the data are shared generally and then through the African CDC. And this basically gives us an idea of what is circulating. Within Africa, you see we have about 3 major strains or lineages of the virus circulating, lineage A, B1, and then B2, and then that's showing us clearly lineage A, which is the original lineages from Wuhan, and then lineage B1 from Europe, and then lineage B11 from the US, and then that shows you clearly, the kind of relationship, talk about movement of people. And in these most cases in Africa, or virtually all cases that were introduced to Africa were imported from all these destinations. But then how do we use, genome sequencing to understand, how the virus is evolving and is moving across the continent? I'll just give you 3 examples, for instance, how we've been able to work, to use genomics as a way to shed light, into how the virus is evolving. The case in Kenya, for instance, early in the outbreak, samples collected between Kenya, and then between other areas of the country show evidence of imported cases, but also evidence of community transmission, especially between, Nairobi and then some coastal regions of Kenya. So, and this has been very helpful in order to enable, public health authorities actually to put down the necessary countermeasures. So, through these kind of systems, the government takes the appropriate measure. The second case is a case of Nigeria, for instance, where very early, the first few cases of, in the state of Osun State in Nigeria, we showed that it was community transmission between a cluster of people probably traveling together. And that was very helpful because they actually blocked them, and then they were able to contain within that village, they were able to contain the outbreak. From that time up to now, that particular enclave have not shown a single case. The third example is a case of South Africa, for instance, where the South African, South African Genomic Surveillance Network, has been able to use analysis technology and to characterize an epidemic or an outbreak of SARS-CoV-2 in 3 major, what I call metropolitan area of South Africa, the Gauteng, and Pretoria, and then the KwaZulu-Natal. So they were able to show that, the reproductive rate was very high at the time where, this is about a month ago. So, and then they also showed that these were, the virus was introduced in the country, predominantly to Europe, and then also to show that, the infections, were characterized the infection, and then these were mostly acquired through nosocomial infections, in a major hospital in Durban, for instance, and then transmitted from that hospital to the neighborhood and other areas. So, and this was also associated with some kind of showing evidence of increased death rate and with some other polymorphism that they observe. Oh gosh. That's like a move. Oops. So we also use this as a way to monitor, viral evolution and then spread on the continent. So this basically gives you an idea of how, you can see here in the small box, which is the Africa the Africa map versus the Nigeria map. But then that shows you again what I've mentioned before, that these lineages from Africa are clustering. It shows not only importation, but at this point, if this was made big, you could also see that there's actually transmission within African countries, and then this is due to population migration. We also look into how the virus is evolving. We investigate the presence of the spikes, the mutation in the spike protein, and we found, for instance, in the case of Nigeria, for instance, 4 patients. And what was very interesting about these 4 patients that we sequenced the samples and we observed 4 mutation. This is exactly the D614G mutation in the spike protein. And what was interesting about it is that when we went back to the clinician to find the information about this patient, we realized that these 3 of them actually had severe disease, and very interesting. So, that shows us exactly that we have this mutation. But monitoring this mutation becomes very critical, because we also identify in another study, in Ghana described the presence of mutation in the nucleoplastic protein, and then we all know that the presence of mutation in this particular protein could lead absolutely to false negative as the PCR is concerned. So we're using all of this sequence data and information to harness and to continue to develop the CRISPR diagnosis that we started developing for Lassa fever for COVID-19. And then you're probably going to see the next few slides how we've developed this test and then validated, and it's doing very well, very well. So, and as you all know, I don't need to teach you, we leveraging gene editing technology as a way to, to use that to develop really point of care diagnostics in the field. And we have successfully done that in the case of Nigeria. We went from developing a Cas13 test that was 1.5 hour long to a single simple assay that is probably more, that is just about 15 to 20 minutes long. So, and then this basically been done in collaboration with colleagues at Harvard and then at the Broad Institute. And with the new assay that we call, it's called SHINE. So this basically gives you an idea of what we've been doing with CRISPR already for a disease called Lassa fever that is endemic in Nigeria and West Africa. And here you can see like a Sherlock lateral flow reads out. So giving you a clear view and how beautiful, this assay is. We have a sensitivity about 86% now, and then our specificity is about 92%. Leveraging, CRISPR-Cas13, we also working on multiplexing these assays, not only for SARS-CoV-2, but also for batteries of pathogens that are circulating within the region, because we have a big project on mapping the microbial landscape across West Africa. And then we've been able to develop a tool called Carmen, and the Carmen has the ability to detect, hundreds of microorganisms in one single assay. So the goal here is working with the Africa CDC and then the other national centers for disease control across the continent, actually to come up with what we call Sentinel. Sentinel is basically, as you understand, is a pathogen detective network that we just set up. But then it's built on using sequencing technology to build the gene-based diagnostic panels and then transform those ones to point of care. So right now, the camera that we have in our lab can do a whole lot. So this is definitely leverages a new initiative called Sentinel, and the Sentinel is built on three major pillars, and that is an early warning system to preempt pandemics. And it is built on three major pillars: detect, connect, and empower. And this basically is a collaboration between my lab and the Broad Institute. Ultimately, the goal is actually to build the next generation of African pathogen hunters. Thank you.
All right, fantastic. Thank you, Christian. We have a few questions here. One is, any connection of people treated for HIV and COVID severity? Do you see any correlations there?
Those that we saw, and then the ones that we saw with severe disease actually did not have, HIV. We had one of them that had diabetes, but the other ones were just people that were normal.
I guess I'd have a question for you, Christian. We've talked a little bit about this before. I mean, it's great that you're not seeing this spread in places like Nigeria, like you're seeing in other parts of the world. I mean, do you think there's an underlying molecular response or a reason for that lack of spread?
Well, I think it is possible. I mean, it could be it's probably due to so many reasons. And I don't think that one single reason might be responsible for the lack of spread. And then you can also see that within Africa, where the case fatality rate is pretty low compared to what happened elsewhere. It might probably be due to the fact that, the genomes of Africans are pretty diverse. I think we have a project looking at it and then trying to relate the genetic diversity of Africans to COVID, to some phenotypic response across the continent. So I think it's also possible that we have some underlying immunity that is based on probably other circulating viruses that probably would have selected the genomes of African a little bit differently, over the years. So I think these are basically just mere speculation. I'm not too sure, but I think, some kind of probably genetic factors may be responsible for what we're seeing. And I think this has to be investigated.
There's one other question here. Can you speak about the viral genetic diversity in Africa compared to the sequences that are coming out of Europe?
Well, I mean, we've seen clearly, as I showed before, that a lot of the sequences. A lot of the largest strain that actually we saw in Africa, which is a B1, actually is the one circulating in Europe, and this is due to the fact that you have a lot of business, and then human migration between Africa and Europe. And the next one is definitely China, and then you also see, the US. So that this is highly explained. But then the rate at which the virus will be evolving in Europe might be different. And also remember that the pandemic in Europe has been there for a while compared to Africa. So I think we basically monitor, and I'm sure in the next few months we should be able to see whether the acceleration of what we see in Africa may be dramatically different or similar to what's happening in Europe or in the US.
One last question. It's kind of similar to a previous question, just about the synergy between COVID-19 and Ebola viral infection. I don't know if there's enough data yet to say that, but I don't know if you have any comments on that.
I'm not too sure because I know that during the outbreak, we had a resurgence or a new outbreak in the eastern part of the DRC. So I haven't been in touch with the colleagues at the DRC to say if their cases were or if there's a synergy between both. I think it is something that we'll probably have to see as the DRC is battling with these two beasts at the same time.
Okay, thanks, Christian. Fascinating stuff. Great. So we're going to jump back to Davey at this point. We're going to see if we can. Davey, do you want to come on and share your slides?
Yeah, I'm going to be talking about the ongoing program of developing the antiviral remdesivir for COVID-19. And just given a quick background that it's a nucleotide analog with activity against several RNA virus families. And it's an IV medication, not suitable for oral administration because of the first pass metabolism, but we are working on an inhaled dosage form of remdesivir, which should be in the clinic imminently. And here we have the Rhesus macaque data from a model of SARS-CoV-2 infection. The animals were dosed with either the IV remdesivir or vehicle. And the clinical score results suggested that the treated group had a clinical benefit. And this was also confirmed on lung pathology. So most of the time, most of this talk will be spent on the clinical trial results from the remdesivir program, in particular these first three. So the two Gilead-sponsored trials asked the question of whether the 5-day course of remdesivir was as safe and effective as a 10-day course, and the NIAID ACTT trial was a placebo-controlled trial aimed at asking the question of whether remdesivir provides efficacy over placebo. And there are now two global WHO-sponsored studies of remdesivir around the world, and we'll talk briefly about the next step in combination therapy. There were 2 trials initiated in China, one in severe patients and one in moderate patients. And because the outbreak was controlled fairly quickly, the enrollment was not adequate to answer the primary question of efficacy in the severe study, and the moderate study was suspended also due to the low enrollment. So I think we're here when we lost the Wi-Fi last time, and this is a timeline to demonstrate kind of the incredible pace of activity in the remdesivir program, and I think also kind of speaks to something that Nevin was talking about earlier on, which is what we can do when we all come together and work towards ending something as drastic as COVID-19. So the first compassionate use request for remdesivir at Gilead came in on February 25th, and a few weeks later, the Gilead simple studies are named as such because they were aimed at answering the question of 5 versus 10 days of remdesivir. Those launched a few weeks later, and we also then initiated an expanded access program as we recognized the exploding need for remdesivir around the world. And on May 2nd, we were granted an emergency use authorization by the FDA, and on May 8th, the drug was given full approval in Japan. And at the same time, there were a number of collaborative studies that were also launched, the China studies, as I mentioned, the NIAID trial, and the WHO studies as well. So again, this really represents the coordination of hundreds of people across many different institutions around the world. So to get into some of the data, we'll start with the NIAID Adaptive Treatment Trial, or ACTT. This was published last month in the New England Journal of Medicine, and we'll just look at the main results. The NIAID study was a phase 3 randomized, double-blind, placebo-controlled trial in patients who were confirmed to have SARS-CoV-2 and pneumonia and an oxygen saturation of less than 94% on room air, or requiring supplemental oxygen, or people who were mechanically ventilated were also enrolled in this study. So in this first part of the study, about 1,000 patients were enrolled and randomized to receive either 10 days of remdesivir or placebo, with the primary outcome being time to recovery. Patients were enrolled from February to April, and on April 27th, the Data Safety Monitoring Board recommended that the preliminary primary data be provided to trial team members, and we'll take a look at what drove that decision. So looking at the primary outcome of time to recovery, the remdesivir group had a shorter time to recovery compared to the placebo group, 11 versus 15 days, and this reached statistical significance. There was also a trend for a mortality benefit of about 7% versus 12%, but this part of the study was not fully powered to answer the question of mortality. There was also a key secondary outcome, which was that the odds of clinical improvement were higher for the remdesivir group versus placebo at day 15. And interestingly, in this study, actually, this key secondary outcome had originally been the primary endpoint, but as the understanding of the disease was evolving kind of at the same time as these large trials were being launched, it became increasingly recognized that time to recovery would probably be a more meaningful endpoint. Now to look at the two, So next we'll take a look at the results of the two Gilead simple trials, one in severe patients and one in moderate patients. The study in severe patients was also published last month, and again, this was aimed at asking the question of whether a 5-day treatment would be similar to a 10-day treatment. The advantage of a 5-day treatment course would be that we would have the potential to essentially double the global drug supply if if 5 days was adequate for patients. So this study was a phase 3 open label trial. And again, we had trial sites around the world. Part A of the study was the main part of the study that informed the primary efficacy endpoint. And here we enrolled people greater than 12 years of age hospitalized with SARS-CoV-2, and they had to have an oxygen saturation of less than 94% or be requiring supplemental oxygen. But in Part A, we did not enroll patients who were intubated. And so we'll be focusing on that population for these results. We did enroll people who were mechanically ventilated in Part B, and that was kind of an extension cohort where we enrolled several thousand more patients, and those data will be reported descriptively at a later time point. But the primary endpoint of Part A was the clinical status as assessed by an ordinal scale on day 14. So the primary endpoint was assessed by looking at the odds ratio for clinical improvement and comparing the 10 to the 5-day group. And after adjusting for the baseline disease severity, we found that the odds ratio was 0.75, which indicates that there was no difference in efficacy between 10 and 5 days. This was important it's important to note that this result was seen after we adjusted for baseline disease severity. It turns out, although this was a randomized trial, more patients who had severe disease status actually ended up being randomized into the 10-day group. And that really drove a lot of the clinical outcomes at day 14. So after we adjusted for those differences, the 5 and 10-day group looked very similar. But we did want to ask the question, okay, 5 days seems to be similar and adequate for the overall population, but are there some patients who could benefit from an additional 5 days? So to do that, we recategorized the patients based on their oxygen support level at day 5. So you can see the different disease categories or levels of oxygen support listed here across the x-axis, and the oxygen support level at day 14 on the y-axis, and then each color represents a different outcome. So when you look at the group of patients who had progressed to mechanical ventilation by day 5, you'll see that the 10-day group actually had a lower mortality rate compared to the 5-day group. So that suggested that in those most critically ill patients, a total of 10 days of remdesivir may actually be beneficial. And that then informed the language in the emergency use authorization granted by the FDA. Now, the numbers are small in this subgroup analysis, so it remains to be confirmed, but the EUA, the emergency use authorization, does suggest 10 days of remdesivir for people who are mechanically ventilated. We conducted a similar study in patients with moderate COVID-19, and this was defined by an oxygen saturation greater than 94% on room air, and but these patients were also hospitalized. So in Part A of this study, 600 patients were randomized in equal proportions into groups receiving either 10 days of remdesivir, 5 days of remdesivir, or continuing their standard of care. And of course, the standard of care was quite variable at the time of this study around the world. I think standard of care is still being defined for this disease, but it was nice to have a comparator in this moderate study, and then ask the question, what are the outcomes, the clinical status on day 11 for each of these groups. We also have a Part B to this study where we have enrolled up to an additional 1,000 patients. these results haven't been published yet, but the preliminary results were announced in a press release in early June. And what they suggest is that when you look at the 5-day remdesivir treatment arm compared to standard of care, there's an odds ratio of 1.65. This suggests that there's a benefit, a statistically significant benefit for that 5-day remdesivir treatment compared to patients who got standard of care. And the 10-day compared to standard of care also had a trend towards benefit. And we're doing some additional post-hoc analyses to understand some of these differences further and hope to publish results in a few weeks. So to summarize, let's take a look at where all of these studies have kind of led us in the understanding of the role of remdesivir along the spectrum of COVID-19 disease severity. If you start with the mild to moderate population, now in mild patients who have not progressed to the point of needing hospitalization, could there be a benefit for outpatient treatment with either IV remdesivir or inhaled remdesivir? You can imagine a situation, for example, in a high-risk population, such as long-term assisted living facilities. If there is an outbreak, an IV form of medication could still be useful. But of course, inhaled, I think, would lead to a significant advantage in mild, non-hospitalized patients, but we are currently investigating that. And then we looked at the moderate results just now. So those results suggest that the 5-day course of IV remdesivir provides a superior clinical improvement compared to standard of care. And then moving into the population of patients who have severe COVID-19 as defined by a requirement for supplemental oxygen, the NIAID trial showed us that 10 days of IV remdesivir compared to placebo leads to a shorter time to recovery. And the Gilead-sponsored 5 versus 10-day trial showed similar efficacy between those two treatment courses. And in mechanically ventilated patients, as we saw in that subgroup analysis, 10 days of remdesivir may be beneficial. But a big question that remains is whether some type of combination treatment could be used to further reduce mortality. And there's an increasing understanding of potentially a dysregulated immune response that might be contributing to the excessive mortality in COVID-19. So 2 phase 3 trials are now kicking off, one combining remdesivir with a JAK inhibitor, baricitinib, and that's in the next part of the NIAID ACTT trial. That'll enroll 1,000 patients randomized to either the combination or remdesivir alone. And then a trial that we're collaborating with Roche on is the Remdacta trial, which combines remdesivir with tocilizumab, an IL-6 inhibitor. So just to finish up, in addition to these trials and the donation of 1.5 million doses, Gilead is also really working hard to expand access to the drug, and we recently entered into long-term licensing agreements with 5 generic companies in South Asia, and are also in advanced discussions with UCSF to work on how to make this drug accessible around the world. So with that, I'd like to thank you for listening, and thanks for your patience with the technical difficulties, and really extend our deepest gratitude to the patients who have made all of these studies possible, and their families, and all of the scientific community around the world. So thanks, everybody, and I'm happy to take questions, Nevin, if there are any.
Yeah, fantastic, Devi. Really exciting stuff. There are a few. We'll get to a couple here. Does remdesivir have different effects in patients with early disease versus those with advanced disease? What's the take-home message there?
So it does seem that the earlier you can start treatment with remdesivir in the disease course, the more beneficial it likely is. We didn't get to that in these data, but we have some sub-analyses suggesting that if somebody has a shorter duration of symptoms and they initiate remdesivir, they have a better chance of a good outcome. So again, we're working on that inhaled formulation to try to make that more feasible.
There are a couple questions here also talking about combinatorials with other FDA-approved drugs. You alluded to some of this with the JAK inhibitor and the work with Roche. Is there more efforts along those lines that are being explored in the context of clinical trials?
So we, we're exploring a lot of different options, and I think there's a lot of interest in the scientific community to see what the optimal combination could be. And I know that recently there's been some excitement about dexamethasone, for example. So I think the door is pretty wide open. We have these 2 collaborative studies that we're taking part in now, but I think we're open to others as well.
Maybe one last question here. Any different effect on remdesivir on the different groups of SARS-CoV-2? Have you looked at that?
I guess I'm trying to understand that question. The different groups of, I guess, the different
clades that are being identified.
Different clades.
Yes.
So we have the global trials, and we are trying to collect samples to do that type of further analysis. I think these trials were launched on such a short time scale that doing that type of extensive additional sample collection has been a little bit challenging, but we're definitely interested in that question as well. So we don't have an answer yet.
Okay, very good. Exciting stuff. Thank you, David. I'd encourage you to answer some of these questions also in the chat box if you could. So we'll transition next to Danielle Swaney. She is an assistant professor in the Department of Cellular and Molecular Pharmacology at UCSF. She's also affiliated with the Gladstone Institute here at UCSF, and she's going to be discussing the global phosphorylation landscape of SARS-CoV-2 infection.
Okay, thank you, Nevin. Yeah, so this is work done in as part of the QCRG efforts. And I just want to start by acknowledging that this is a huge team effort, about a dozen labs and over 50 different authors. And so when I use the term we in this presentation, it's a collective we of many different people contributing, and I've tried to highlight those people throughout the presentation. And so I don't think I need to convince anyone that studying the biology of SARS-CoV-2 infection is important, but I just want to highlight that phosphorylation plays a very important role in viral infection. So here's a few figures from a review on influenza virus infection that kind of highlights that different modes of phosphorylation signaling, receptor tyrosine kinases, PI3 kinase, ERK, many other pathways and kinases are involved in the entire viral life cycle from attachment, entry, and release. So phosphorylation plays a very critical role in viral biology. And this is not a coincidence. As opposed to transcription and translation, phosphorylation signaling represents a very rapid mechanism to manipulate host cell biology. So within a second, you can easily add a phosphorylation onto a substrate and turn a kinase on, and then be able to propagate that signal through many of its downstream substrates. And so it's that this is a way for viruses to rapidly manipulate the host to their advantage, but it's also a way for the host to rapidly combat viral infection. And so studying phosphorylation represents a very important part of understanding viral biology. And so in our lab, we've been using phosphoproteomics to understand phosphorylation signaling. And so our general approach is to perform global mapping of phosphorylation changes, and then use bioinformatics approaches to translate these complicated phosphorylation information into more manageable kinase activity regulation. And as kinases represent a large fraction of the drug targets, we can then use this to prioritize drugs with antiviral efficacy. So that's our general approach to this project. And so the experimental design we used is we started with Vero E6 cells, which are African green monkey cells that are highly susceptible to SARS-CoV-2 infection. And so we either perform SARS-CoV-2 infection or a macaque infection, and we incubate the virus with the cells for 1 hour, and then collect cells at 6 different time points from 0 hours to 24 hours after infection, along with paired macaque time point infection at 0 and 24 hours. And then for every condition and time point, we collect this in biological triplicate. And for each sample, we perform a phosphorylation analysis as well as measure protein abundance differences upon infection. And here we've used a data-independent acquisition mass spec approach for our quantitation. And because we're using Vero E6 cells, we then perform alignment of all protein and phosphorylation sites to their autologous human proteins. And so as two measures of the quality of our data, in the protein abundance analysis, we're able to measure the individual proteins derived from SARS-CoV-2, and we can see robust induction of those viral proteins, primarily after 8 hours of infection. So we know we're getting good infection. And then looking at our the quality of our quantitation, we observe high correlation coefficients between samples within the same conditions, usually 0.95 or above. So we have high quantitative reproducibility. When looking at those proteins that are statistically different upon infection, we find that only approximately 160 proteins are significantly regulated upon infection. However, if we look at phosphorylation sites, now we've seen approximately 10-fold more phosphorylation sites than we do proteins that are significantly different between these upon infection. So from this, we can suggest that phosphorylation signaling represents primary host response, as we see many more differentially regulated species here. So the phosphorylation signaling changes represent thousands of different phosphorylation sites, and understanding the biology of this complex number of phosphorylation sites can be difficult. So here we turn to our collaborator, Pedro Beltrao, who's developed this tool called Phosphate to help make this data more translatable and interpretable. So in this Phosphate tool, it has a database of millions of different phosphopeptide quantifications over hundreds of different conditions, and then also has a database of known kinase substrate interactions. And so with this tool, you can input your phosphorylation data, which represents phosphorylation changes on substrates, and it will aggregate that information into the known kinases and generate a score that represents the change in the activity of the kinase based on its substrates. So using this tool, here we have a subset of our activated or inactivated kinases. And you can see those in red are the kinases that are activated upon infection, and those in blue are the ones that are inactivated. And we see many well-studied kinases such as PIK3CA, AKT, for example. But one kinase that we are particularly interested in here was the casein kinase 2. This is a heteromeric kinase. So here are two components of this holoenzyme here that are both rapidly activated upon SARS-CoV-2 infection, and this activity is sustained through 24-hour time course. And one reason we were particularly interested in casein kinase 2 is from our previous protein interaction studies. So when we performed protein interaction studies in SARS-CoV-2, we observed that the N protein from SARS-CoV-2 interacted with two components of casein kinase 2, as shown here. And meanwhile, as I just showed you, infection experiments showed increase in CK2 activity based on many substrates that had increased levels of phosphorylation. And several of these substrates are known to be involved in cytoskeletal reorganization. And shown here on the right is just some of these different substrates and their specific sites of phosphorylation, where you can see that upregulation. So based on this information, we wanted to look at how, if N protein might be influencing cytoskeletal regulation in SARS-CoV-2 infection. And for this, we took an immunofluorescence-based microscopy-based approach. So here we have immunofluorescence in CACO2 cells, where we have F-actin represented in white. This is a cytoskeletal protein, and our SARS-CoV-2 protein in red, and then casein kinase 2 in green. And so if we zoom in this white box here, that's shown here, and you can see this filopodial protrusion from the F-actin staining here. And quantification of these protrusions shows that it's significantly increased in frequency upon SARS-CoV-2 infection as opposed to mock infection. And then if we zoom in even further, we can look at the co-localization of N protein and CK2. And so here you can see co-localization of these two proteins, and we find that approximately 40% of the time, N and CK2 are co-localized. So, and to look into this even further, we collaborated with Elizabeth Fisher, the next presenter in this symposium, who performed transmission electron microscopy. And so now we can zoom in even further and look at these protrusions. And the interesting thing that we find here is that we see what appear to be viral particles that are budding from these protrusions. So we hypothesized that N protein is promoting CK2 activity, and this is part of the process of viral budding and egress. So to try to understand the role of N specifically in this process a little more, we performed another phosphoproteomics experiment using the same approach as before, but here we, instead of a viral infection, here we just induce N protein in a doxycycline inducible setting, or no induction of N protein. And then we use our kinase activity analysis again, and here we can see that N protein expression alone is sufficient to drive increased CK2 activity, but also causes downregulation of other kinases such as macaque and CDK2, or excuse me, and CDK. So from this data, we found that N protein physically interacts with several components of CK2, and this leads to activation of CK2. And so the next step is to try to identify drugs that we may be able to use to target this interaction and increase in activity. And so in collaboration with Kayvon Shokat's lab, we mapped drugs to our differentially regulated kinases, and one drug that came up that targets both several members of the CK2 kinase is silmitiricib, which is in phase 2 clinical trials and is currently being considered for human testing. And so we did infection assays in Vero E6 cells, and we found that this drug has antiviral activities with an IC50 of 2.42 micromolar. And so this seems to be an effective way of inhibiting viral replication. We performed this type of mapping between kinase inhibitors and kinases for our entire data set, and identified several other drugs that also had potent antiviral activity at submicromolar concentrations. And in total, we were able to map 87 drugs to these dysregulated kinases and pathways. 68 of these drugs have been tested in our study, which is currently under review. And 38 of these drugs, we're happy to find, have IC50s that are well below their cellular toxicity values, suggesting they may be valuable inhibitors. So in summary, we used a phosphoproteomics approach of SARS-CoV-2 infection to identify dysregulated pathways and kinases upon infection, and we find that SARS-CoV-2 is able to dramatically rewire host signaling, and this causes activation of CK2 and localization at the viral-induced filopodial protrusions, where it seems that the virus is budding from. And we can use this data then to prioritize drugs that target these dysregulated kinases and pathways. So in closing, I just want to acknowledge again, this is a large team effort, and I wanted to thank the QCRG, QBI, and Krogan Lab, and the Gladstone Institute Institute for their support, as well as our funding from DARPA and the NIH.
All right, fantastic, Danielle. Thank you very much. A few questions here. Do you think casein kinase 2 is affecting the interaction of N with the viral RNA itself?
Oh, good question. We don't know the answer to that.
Okay. What about Davide Ruggiero? What about phosphatases? Does the virus manipulate phosphatases? Did any of them come in the interaction map? Do we have any evidence that they're being hijacked and rewired as well?
We haven't really looked into the phosphatase data, so I don't know the answer to that either.
Okay, so another question. This is very cool data. It was shown that casein kinase 2 is needed for listeria spread too, where it phosphorylated WAVE and WASP. Do you see WASP and WAVE being differentially phosphorylated in infected cells as well?
I don't think we observed this.
Okay, and is casein kinase 2 activation also observed or been reported for SARS-CoV-1 or MERS? That's a good question.
It has been implicated in another virus, but I don't think it was SARS-CoV-1. I can't remember.
Okay, and maybe one last question here. The cell cycle or the CDK inhibitor you alluded to, it targets many CDKs, including CDK1 and 2 and 9 and 12. Is there a way to distinguish which ones you think are being misregulated in infected state?
I mean, our data shows CDK1 and 2 as being inactivated, but there may be others beyond that.
Okay, very good. All right, thank you very much, Danielle. And again, I'd encourage you to look at the chat box, please, and answer any of these outstanding questions. And we are going to now segue to the last speaker of the session, Elizabeth Fisher. She is in Rocky Mountain Labs in Montana, close to where I'm born and raised in Saskatchewan, very close. So she's the chief of the RML microscopy unit at the NIAID. And she is going to be telling us about some very cool imaging projects, not just with coronaviruses, but with other infectious agents as well.
Hi, thank you, Nevin. I just want to echo what we've heard several times before. I think one of the silver linings of this pandemic is really the opportunity to collaborate and cross a lot of different boundaries culturally as well as interdisciplinary. So it's really great to have this opportunity to share some things with you that we've looked at in the study of pathogens. So like Nevin said, I'm not at the mothership in Bethesda, but we're about 2,322.4 miles away with a population density of about 1 million people. So social distancing is not a stranger to us. The Rocky Mountain Laboratories sits in a beautiful Bitterroot Valley. It's a 33-acre campus that actually had its roots in Rocky Mountain spotted fever. About a decade ago, a BSL-4 lab was added, the Laboratory of Virology, led by Dr. Heinz Feldman. And we study a variety of things with them, including the coronaviruses that are causing havoc today. My group is a core facility. We run microscopy. We have a variety of different tools and technologies to try and address the variety of pathogenic questions that we're asked. So we have SEMs that look at the surface details of things, TEMs that look at the cross sections of things. We can do focused ion beam to get 3D volumes, and of course we have cryo-EM as well. RML's SARS-CoV-2 timeline kind of started back in December when the Wuhan virus became available or known, I think the Wuhan pneumonia. The Laboratory of Virology came up with a plan of attack. They were heavily involved with the Ebola outbreak and basically had everything kind of ready to go for a new pandemic. So the first sequence came out early. Things started being identified with the ACE2 receptor, who declared it a pandemic. We received our first isolate here from the CDC back in early February, and from there we started generating some images that have been used by the public. On Valentine's Day, the inoculation of the Rhesus macaques was administered, and that was to really start to identify an animal model that could be used to study treatments and vaccines. remdesivir was studied here. We've got aerosol surface stabilities, and then a variety of vaccine studies. Just with NIAID alone, between here and the main campus, there's been a number of publications that have come out really quickly. And again, this is obviously a global thing as well, so this extends well beyond NIAID and in collaboration with many people. The study that we had with the animal models was with Dr. Vincent Munster and Emmy DeWitt, and looking at the lung tissue at a 3-day monkey, you can see this area of pneumonia starting to develop. And if you look at a normal lung tissue, this was actually 7 days post-infection, but it kind of this more normal spongy area that you'd predict. You have a lot of wide open space, and basically it looks fairly normal. But in that region where there seems to be a lot of infiltrate, you see a lot of edema, a lot of swelling, collagen, fibrin. You can start seeing different kinds of monocytes and inflammatory cells in response, and we were being able to identify a virus that was in the actually type 1 pneumocytes. The important thing about doing animal studies too, especially when you start thinking about drug delivery, is what kind of environment would this reside in. So it's actually important to do things beyond the cell culture, of course. Here we're looking at Mycobacterium tuberculosis infected rabbit lung tissue, and this is just to illustrate that the bacteria reside actually in these big lipid droplets. And so it's important, again, when you're studying, just to be aware of what that residence is in the natural infection and host. We have several ongoing studies now with cell biology. Obviously, when you're studying, as Neva said at the beginning, one thing that is really important is to understand the different pathways within a cell, because this will give you good targets for treatments or vaccines. And RNA viruses themselves tend to really rely heavily and exclusively on cell membranes, and they're famous for having mass rearrangements of cell membranes within cells once they infect for replication success. And so this can be advantageous to the virus because it can give you kind of a high local concentration of viral products within the cell. It can give you a scaffold for spatial organization, so things are ready to go together. And it can also provide some protection from cell responses. And so when you look at these, there's a lot of different things you can see. So you can look at things that looks like ribosomes, so maybe the ER is somehow implicated. People talk about autophagy. These are mitochondria that look like they've been pretty heavily damaged. There's sometimes more single and double cell membrane things that might be happening. You can also see little viral protrusions and spherules starting to develop. And so this, for us, asks a lot of questions. What actually is involved with this mechanism? As Danielle just mentioned, we worked with Nevin and group on looking a little bit at the SARS-CoV-2 eruption off the cell surface. You can also see it on the flatter part of the plasma membrane, but certainly you see loads of it coming off the filopodia as well. And so this starts asking the question, how exactly are things attaching, getting in, doing their thing inside the cell and getting back out? And I just want to give a shout out to our visual medical arts department. They've done a wonderful job colorizing our images and kind of bringing them to life. Some of these you've probably seen in the public. Danielle kind of went over that overall replication cycle, but again, you have some mechanism of entry, you have some kind of membrane-bound compartments, somehow things assemble, and somehow they get back out. And there's a lot of different steps involved, and it was a really great talk on the last section, learning about some of the phosphokinetics. In our group, we often look at things and turn to immune labeling to try to dissect what's happening within the cell. And I just want to point out, these slides came from George Posterman at Utrecht years ago, but I still like to use them. So we may have a section or a sample sitting on a grid. We have the antigen of interest. We have an antibody that combines with that. And then we have some kind of electron dense probe that we're able to visualize in the microscope. If you have specific labeling, you have very nice punctate labeling on a antigen. If you have background labeling, things don't look so great, and you have to be careful in your interpretation sometimes of what you're calling real labeling. This is what some of that might look like. On the top, you're seeing some labeling of Golgi compartments. We can distinguish trans as well as cis Golgi. We can do correlative techniques where we can identify rare events and then do TM or SEM to look at those in more detail and correlate. Or we can do different peroxidase stainings to be specific about what's host and what's viral antigen or bacterial. So to give you an example of how we might use that in a viral infection, this is a poliovirus study that we did several years ago with Dr. George Belov and Ellie Ehrenfeld. And here again, like in the positive RNA viruses, you get this collapse of membranes, and they're taken over and produce these little replication complexes. Polio has been studied for decades and decades, and along the way, there's been a lot of controversy about single cell membrane things, double cell membrane, or double membrane structures, and what's actually real. We did a big time course actually and found that that both people were right. It was kind of dependent on where you were in the cycle. We were able to label for RNA synthesis and kind of looked at those rounder structures. We found Golgi involvement. Calnexin labels the ER. We actually saw the labeling here, but the replication complexes are up here. So the labeling didn't actually seem to be suggestive of ER at that time point. When we looked at it by tomography in 3 dimensions, again, we saw sort of a time course between 6 and 12 hours, where you start with a single cell membrane, but then you get this interconnected complex that is actually part of that replication complex. This is kind of give you an example of cryo-electron microscopy. Nevin talked about a little bit of work they're doing back at UCSF with this too, with their integrated facility, but you can flash freeze a sample. So proteins of interest or virus or bacteria, they're frozen very cold, very quickly, so the ice looks like glass. You put them in the electron microscope and start taking images, and you can take thousands and thousands of images and get viruses or your protein of interest in all different orientations. You can then take those pictures and back project them and ask what kind of orientation would have made this shadow. And when you start doing that, you start collecting these things in higher resolution. You're able to average those images together. So once you get your initial models, you can start feeding back in more images and then start building on that. And in the end, come up with a fairly high resolution model of what you're looking at. Let's skip ahead. So we did that with poliovirus. This is actually quite old, so the detectors are a bit older back then. It's not super high resolution, but here we have poliovirus that's been frozen in ice. We have neutralizing antibodies on the surface. And we can take a look at those neutralizing antibodies, and then after reconstructing them, dock the virus in and have the virus behind it, but dock the antibody in. And we can look and see with these 2 different types of poliovirus where these neutralizing antibodies actually bind. And when you understand that, it gives you an idea, especially if one antibody or vaccine might be more neutralizing than another, what the difference is. So you can see in the dark blue here, the attachment sites of the neutralizing antibody, how they rest deep inside these canyons on the surface of the virus, but with different degrees, which gave us insight into why one was a better binder than the other. You can take a look at that in a mesh network and actually identify the amino acids then that would be involved and obviously be good for studying targets. And just briefly, I want to give you a quick idea of what we do with some of our influenza studies. Influenza, as most of you probably are aware, on their surface coat have hemagglutinin and neuraminidase spikes. And things happen poorly for us when there's an antigenic shift and you have kind of a mixing between maybe pigs, birds, and human strains that we're not used to come up with a new strain. And when this happens, it can actually cause a lot of problems for us. The influenza A strains is one of 4. They're A through D. They're very small, 1,000 times smaller than a human hair, only have about 11 genes, and they need a host to survive and multiply. And obviously, they cause a lot of things like we're seeing today as well. So one of the studies we did was with Peter Tlanda, who was at the time at the NIH. Now he's over in Heidelberg. But we were looking at some viral-like particles and seeing how well they represented the influenza particle themselves. So we looked at distribution of the HA and NA spikes on the surface of this particle. And we looked at the curvature, we looked at the inner matrix, and found that it was actually very similar to wild type. And this was then used to study with these small mini membranes interactions and how they would change the entry of this viral particle when we can arrest it in an entry state. and found out that it was really based on the physical properties of the target membrane. So basically, they were able to see that changing things like cholesterol composition within those membranes would actually change how this virus would actually attach and enter into that potentially a cell or some kind of endosomal compartment. And then further, with John Gallagher and Audrey Harris, Barney Graham at the Vaccine Research Center at NIH came up with a new idea where with influenza, oftentimes the part that, of course, is changing and why we have to get a flu shot every year that's different is the head groups on that hemagglutinin may change. So there's a lot of different types. And so Barney decided to have a new strategy at vaccine design, and he actually looked at using small ferritin nanoparticles and attaching the stem loop region, which is more highly conserved between flu species. And so with this, he was able to develop that, and it's actually been through animal models, has been really highly successful. So there, it's going on, I think it's in phase 3 clinical trials now. And why these structures are important, again, is in vaccine design. Barney also came up with Jason McClellan of the spike protein structure really quickly after all of this first erupted. And of course, that gave us great insight for that. And then Dave Easler out of University of Washington also came up with a very nice study looking at the ACE binding domains and then where the spike protein would be partially open or closed and how that affected entry. So we're using the spike protein structures to look at distribution on the SARS-CoV-2 virus itself, as well as comparing that to MERS and SARS-1 and the NO less virulent strain, the NL63 strain. And then we're also looking at spike distribution in several of the different vaccine candidates that we have ongoing here at Rocky Mountain Labs. So with that, I'll end. I want to acknowledge we've got great support from up above. I've got a lot of team of really great microscopists, and then our collaborators, and again, some of our external collaborators. I want to also mention, here's basically my group. This middle group here is the Visual Medical Arts Department that does all the colorizations of our pictures. And this was an outbreak exhibit that's at the Smithsonian Natural History Museum up through, I think, May of 2021. This picture was taken back in 2018, but it was a little bit prescient in that our world is interconnected more than ever before by global trade and technology and even our viruses. And with that, I'll take any questions.
Great, fantastic. Thank you, Elizabeth. So we have a number here. One from Dave Agard. For SARS-CoV-1 or 2, can you see internal structures within the filopodia that would give more detail on how the virus is assembled in such a narrow space? Can you get to that level of resolution?
Oh boy, I don't know yet. I will try. We haven't done any cryo-sectioning yet for high resolution studies on that. We're just doing our labeling experiments literally this week, trying to identify different parts of the virus and different locales in the cells. So hopefully we'll have some answers in the next month or so.
I'll be excited to see those answers for sure. Do you see the virus on cilia and lung cells?
Cilia and lungs, I haven't. I really have only seen them on the type 1 pneumocytes. It can be a little bit of the proverbial needle in a haystack. Often what we're getting are the biopsies of the lungs, and I think often by the time we get the biopsy, a lot of the tissue's been pretty severely damaged and the viruses have kind of moved on from those areas. But I haven't seen them on the cilia.
And then just one question, kind of a general question, and just the challenges of doing these type of things in a BSL-3. I mean, can you just speak just a little bit about that, how you accomplish this?
Yeah, so our microscope's in a BSL-2 lab, and so we deactivate viruses and do a formalin fixation before they come to us. For the cell studies, they're all fixed for 24 hours with glutaraldehyde and paraformaldehyde as well.
Okay, very good. Thank you, Beth. Fantastic stuff. So before the break, we just have a 5-minute flash talk from one of our sponsors and one of our collaborators, Synthego, and it's going to be done by Kevin Holden, who is the head of synthetic biology at Synthico, and he's just going to be giving us a quick summary on utilizing CRISPR genome engineering platforms for COVID collaborations.
Great. Thanks, Nevin, so much. And thank you to the organizers for allowing us to sponsor. It's been, we've had a great time collaborating with the QBI Coronavirus Research Group. So I'm joined today by my colleague, Jared Carlson-Stevermer. And like Nevin said, we're going to talk to you about how we've been trying to utilize CRISPR to do some of these COVID-19 collaborations. So just a little really quick, some brief background on who Synthico is. We're a Silicon Valley-based biotechnology and tech kind of hybrid company. We were launched about 4 years ago. There's about 250 of us. We're advised by Jennifer Doudna, Matt Porteous, who's down at Stanford University, and also Bill Scarnes and Andrew Whitty, who's actually leading the WHO COVID-19 vaccine development right now. So he's a little busy. We're a commercial and research genome engineering organization, really with the goal to enable standardized CRISPR and genome engineered cells to allow for reproducible large-scale experimental workflows that can speed up discovery times. And so, you know, we've got a couple of thousand customers around the world. Our products and services have now been cited over 250 times in the primary literature. We utilize and ship thousands of reagents and hundreds of cells every month. And so what do we actually do? Well, we build these platforms for high throughput genome engineering. And so we've been able to build a novel architecture that allows us to mass produce the RNA molecules that people use for CRISPR, either in a GMP or a research grade type material. We built these automated high throughput platforms for doing cell line and iPSC engineering at scale. And so we have programs for discovery, screening, and disease modeling, where you can knock out genes or knock in tags or variants. And we've recently started taking part in a NIH program called INDI, where we're hoping to build thousands of standardized engineered iPSC lines for neurodegenerative disease research. And so we have biology, software, hardware research, platform developments. We try to work on a model of doing industry and academic collaborations as well. And so we have these teams focused on exploring gene editing technologies, bioinformatic predictions, and preclinical models now. So we started thinking as the shelter in place happened and coronavirus had its grip on the world, how we could utilize CRISPR genome engineering to help fight COVID-19. And so we took note of this bioRxiv preprint in late March, obviously, which became the very excellent Nature paper by Nevin and his group, and many of you who are here today. And so we started to think that if you wanted to actually validate these interactors that were found in infection studies and maybe validate drug hits, you need to actually go through and perturb each one of these interactors and basically knock them out. And CRISPR is a great technology for doing that. So we were seeking, again, a collaboration that could help us utilize our skills to help in this fight. So we reached out to Nevin, and we began a collaboration essentially to provide gene knockouts for all of the interactors that were identified rapidly and for free that could allow for potential follow-on studies. So I'll let my colleague Jared talk about how that actually happened.
Yeah, so as Kevin mentioned, we kind of jumped on this and wanted to act as quickly as possible. And so there's a lot of conversations at the time about what's the best disease model for actually replicating SARS-CoV-2. And so we rapidly optimized our transvection conditions for CALU3s, CACO2s, A549s, essentially anything that could be potentially relevant. At the same time, we synthesized over 1,000 unique sgRNAs, tested them and validated them against targets that were identified in the interactome library. What's really cool about this is that we actually were capable of using our high throughput platform for all the synthesis as well as editing to go from design to knockout in essentially knockout pools in less than 2 weeks. And what you can see here over on the right-hand side is actually our editing efficiency in terms of the number of alleles edited based off of our ICE tool that we have in both the CACO2 cell lines as well as an A549 ACE2 cell line that the Pasteur Institute was kind enough to provide for us for some of these studies. I'd be remiss if I didn't mention one of our really talented scientists at Synthico, Jennifer Oki, that's done a lot of the work in this kind of gene editing field. We've also identified 38 essential genes through our high throughput screening platforms that we can use for follow-up studies on top of that. Then finally, just on the basis of this collaboration, we've heard a lot about kinases, we've heard a lot about different genetic variants. So because we've had this kind of back and forth with all the partners of QBI and the Coronavirus Research Group, we've actually synthesized an additional 150 sgRNAs that have been synthesized and validated on the basis of this. On top of that, we've also provided these cell pools to different collaborators around the world. And so on top of our high editing efficiency, we've actually seen that this has a functional knockout as well. And this is viral studies done by Chris White at Mount Sinai, where just in the simple case, you can see that knocking out of the ACE2 infectors in both the CACO2s and the A549s drastically decreased the percentage of infection that we were seeing within these cell culture models. And this is a study that we are expanding beyond this and moving on to the whole interactome as well as other identified targets from the group. So one of the real key innovations that we've kind of pushed forward with this is the delivery of screen-ready cells for infection studies. At this point, we've created over 400 unique genomic targets that we have high genome editing efficiency, generally on the order, if you look at that last, of around 80 to 90% of alleles that have actually been edited. Additionally, we've learned that these learnings are transferable over cell lines, essentially meaning that we can accelerate time from hypothesis to assay, or that if we find a brand new cell line or a cell model, we can take all the learnings that we had from the previous interactome studies and move them over. We've also live cell shipped these in replicate plates for ease of use. So instead of having to take from frozen stocks and expand and freeze other ones down, you can actually go from receivings to assays in less than a week. And we just have the stocks back here that if anything goes wrong, we can send those out again and get ready to go. We've also had multiple collaborators that have multiple different assays. And so what you can see on the right is we've sent these cell pools to the Pasteur Institute in Paris, Mount Sinai, Georgia State University. And what this can do is actually reduce the risk of false positives, or you can have different variations. The other thing is that we're also looking for additional collaborators. And so if you have any interest in this, please reach out to us. And we are happy to have this conversation and build the collaboration. And we want to put these in the hands of researchers that can do something with them.
All right, thanks, Jared. So again, I know we're up against a bio break for many of you. So thanks for letting us sponsor. Thank you especially to Jacqueline and Gina for getting us set up with that, and also Nevin for collaborating with us. But if you're interested, please contact us to learn more about any of the products or services or collaborations. We have a webpage dedicated for this. We also have a virtual event we run every Thursday on our own called CRISPR Office Hours. Actually, we had Nevin and Jacqueline on a couple of weeks ago. And we've also, Christian earlier was talking about using the Sherlock technology, the CRISPR-based Sherlock technology for surveillance of coronavirus testing. And we've actually worked with groups at both Mammoth Biosciences and also Sherlock to help them develop the materials that they needed to run these COVID-19 CRISPR-based assays. So again, thank you very much for your time and for allowing us to participate.
All right, great. Thanks, Kevin and Jared. If anyone's interested, obviously, please reach out to them. Very powerful technologies and reagents that are available. So we're going to take a bio break now. We're running a few minutes late. We were supposed to start the second session at 10:15. We're going to put that back to 10:25. Okay, so it'll be almost 20 minutes a bio break. And so Kayvon Shokat will be chairing the second session, and Melanie Ott will be the first speaker. I just wanted to thank all the speakers this morning. It was a fantastic a set of talks, really great stuff, guys. Really exciting. So thank you very much, and we'll see you back in 20 minutes.
First up, we have Melanie Ott, who's the director of the Gladstone Institute of Virology and Immunology and professor in the Department of Medicine here at UCSF. She's going to tell us about direct detection of SARS-CoV-2 by Cas13a in a mobile phone. Melanie, thank you.
Well, thank you so much, Kevin. I'm sorry about the delay. I was on a different link, but here I am, delighted to be here and to tell you about our latest effort in detection of SARS-CoV-2 with a mobile phone. We're sort of continuing the theme that Christian Happy has put forward about surveillance and testing. We all know the situation. We have numbers worldwide of new infections of SARS-CoV-2 climbing. But at the same time, we're opening many of the economies worldwide, including here in the US. We're doing this in the absence of a vaccine and only a limited arsenal of therapeutics currently. So we are left with where we were in February and March when we went into lockdown, which is basically social distancing, masking, and massive testing. So far we have performed about 26 million tests in the US. About a tenth of them were performed here in California. But the estimates are that we might need the same amount per day potentially to really safely open the economies and get surveillance in place. So these tests come in two flavors. One are really random community testing, very much lab-based, how Christian has pointed out with, with RT-PCR or with sequencing. And the other one is more individualized, is really more of a point of care testing where we want to test people who want to enter a building, who want to get a procedure done at a doctor's office or want to go onto a plane. These tests have to be very rapid. They have to be, very easy to administer, and they have to be, in theory, trackable in the way that we immediately know where there is a positive signal or a positive test, and we can, start tracking contacts and isolate them. So I have been thinking about point of care testings about 2 years ago, started thinking about it together with my colleagues, Daniel Fletcher and Jennifer Doudna here in the Bay Area. And we were really thinking around HIV-specific home testing, especially for people who wanted to stop antiretroviral therapy and go into clinical cure trials. The idea was really to combine the two expertise, Jennifer's and Dan's expertise, Jennifer and her CRISPR expertise, and Dan is a bioengineer who has really pioneered the use of mobile phones for diagnostics. And so here's just to recap very briefly what Cas13a can deliver. It is one of the many CRISPR-Cas enzymes. And it is, contrary to Cas9, actually an RNA binding Cas enzyme that when activated with a guide RNA here against SARS-CoV-2 and finding its RNA target becomes activated. And when it becomes activated, it cleaves its target like many other CRISPR enzymes, but it also has collateral RNase activity that can be harnessed via a reporter system. And that was first described in 2016 by Jennifer's group. And so when you provide the activated complex with a simple reporter that is basically an RNA bound with a quencher and a fluorophore, and when this reporter is being cleaved by Cas13a, it starts to fluoresce and it can be measured in the plate reader. And this measurement is directly correlating with the amount of viral RNA that you have in your sample. So the initial data by Jennifer's lab showed that this is a very specific assay. So if you use the 3 different guides against phage, you see that you can detect the concentration of the target very well. However, if you take an enzyme that is mutated or you take you supply an off target in the reaction, you don't see any reaction. So this specificity is very good. RNase A is always the positive control for your reporter cleavage, but the sensitivity was not very good. You could see that when you go down in your limit of detection assays, you go into the picomolar range, which is about 10 to the 5th copies per microliter. So this is why 2 years later, Sherlock was developed, which is an assay that relies on amplification of the target. So instead of using the RNA directly, the RNA is reverse transcribed, amplified isothermically, and then transcribed back into RNA for the detection of by Cas13a. And then, as you saw in Christian's talk, for the point of care ease, you use often lateral flow assays to detect very much like in a pregnancy test, whether the test is positive or negative. The advantage is really enhancement of sensitivity, but it adds time because you need to do the amplification. It adds additional reagents and it adds additional variables in your step because you have a lot more enzymatic reactions going on than just the simple direct detection assay. And it loses its the ability to be quantified because through the amplification, you cannot directly correlate anymore the amount of RNA in the target with the cleavage by the reporter. So when we started this, we said we would like to not do the amplification and really stick with a direct detection approach by by just systematically optimizing all components in the assay. And I'll show you a few things in a minute. But also by not using the lateral flow assay, which takes away sensitivity, but by sticking with the fluorescence and reading it on a mobile phone. And really what the mobile phone adds is that we can use fluorescence in an outread. It allows direct detection, so we can be fast and quantitative. The mobile phone obviously is portable, and it has GPS capability, so it's trackable because the results can be immediately uploaded into the cloud. And then what is important in the current state is that it is an existing device and widely available, so you don't have to deal with now massively upscaling manufacturing and getting the parts and running short of them. So when in January, the sequence for SARS-CoV-2 was available, we shifted the assay and we started to generate specific guides RNAs across the genome to see whether we would pick up any signal. And this is really a typical sort of curve that you can pick up in the plate reader over 120 minutes, 2 hours, where you see in blue basically the control, which is just the RNP alone, and we want this to be very flat and not have any activity by itself. And then in purple you see the reaction plus the target, where you want, of course, a rapid detection of the target by the specific guide RNA. And you can see that, roughly half of the guide RNAs gave us some signal, some with better sensitivity, some with less, and then some with more background and some with less. And so this is actually something that we're building on the robot and is done automatically. Then you can either read the slope of this curve as an outread, or you can take an endpoint at a specific time, 30 minutes, 60 minutes, or an hour, 2 hours, and determine whether you have a positive or a negative signal. And you can do this on different targets. You start usually with a synthetic target, which is exactly matching your guide RNA. You go into bigger targets, which is sort of a broader piece of the virus in vitro transcribed, or you use the full-length SARS-CoV-2 virus grown in the BSL-3. And you can see that this is an endpoint analysis here. You can see that the different guides give us different results, but in general we can find out which guide is working and which guide is not working. And then a very important control is just the RNP alone, where we see how much background we have and how much really specific activation we can get out of a guide. So when we repeat that assay now in LOD and limit of detection studies, we run into exactly the same limit of detection that we have seen before in the paper by Jennifer. In about 10 to the 5th copies per microliter, we have a nice signal, but then we start to lose it over time if we use individual guides. Now, when we start combining the guides, we boost the sensitivity, in some cases dramatically, so that we can, with even 2 guides, with only 2 guides, can get into the range of the limit of detection of 1 to 10 copies per microliter in an endpoint, or 10 to 100 copies in a very rapid slope detection that we can do. Now, this combination is not coming at the expense of the specificity, because when we test the assay now on other targets, like for example MERS or just cellular background RNA, you can see that we don't pick up the signal with either the guide alone or the combination of the guide. So the specificity maintains very high. But we also, with combinations, can gain very good sensitivity and specificity when we go into patient samples. Here, patient nasopharyngeal swabs that we obtained from the BioHub. You can see that we very early in this slope can make a decision whether a sample is positive or negative versus our 2 negative controls down here, which are the RNP alone or the negative swab. You might see that the negative swab and the RNP are slightly different in the background. And this is actually where currently most of our focus is, because we know that something in the nasal swab, and we suspect is the microbiome in the nose, is in some cases cross-reacting with some of our guide RNAs. And we're having a massive effort together with our colleague Katie Pollard currently at the Gladstone to rule out any overlap of the guide RNAs with our with the metagenomics data of microbiome in the bacteria in the or viruses in the nose. So we're focusing very much on on this very early step here to eliminate any cross-reactivity, but also to make the nasal swab immediately available, and the RNA on the nasal swab immediately available to go into the assay without RNA isolation. And we're also working very carefully on combining the best guides with the lowest background to enhance further our sensitivity here. But our most interesting weapon in this fight against the virus here has really become the mobile phone. And I'm sort of replacing my colleague Dan Fletcher here, and I'm trying to give you the same expertise in the bioengineering that he has. But I think he really started very early, with really using mobile phones as diagnostics, and initially really as a microscope. It's called the CellScope technology here with an old Nokia phone, where if you add to the lens a magnifier, you can really get into very sensitive images of cells or parasites or others. This has morphed into a clinically applied instrument that is currently used in Cameroon to diagnose river blindness. And you see that CellScope here in the middle with an old iPhone, where basically a blood sample will be introduced into the box, and just by imaging, the phone will decide whether they can detect lower microfilaria in the blood based on their movement actually, and call a positive or a negative within 3 minutes. And this immediately can allow the clinic to decide whether they start treatment or they don't start treatment. Now this CellScope has morphed now into our prototype COVID scope, where we don't really look at images, but we use the lens as a plate reader to read the fluorescence. And so it has a lot of different components, but it still is sort of the same principle that you put the phone on top and you read here in 4 channels with our positives and all our negatives, the fluorescence that we introduce here. And you can see on the right that the curves are pretty similar to what I've shown you before with the single and the doubles, and also in the time frame that we have been. So in general, we thought that the phone has to get the plate reader would be our 10 times better than the plate reader in reading the Google. And mainly what we find is that very much enhances our about 10 times lower levels than what we read with the plate reader. And so in our final attempts here, we are working really to increase our detection limit here in the first 10 minutes of our reading on the COVID scope. And we working continuously to sort of push this limit further down by multiplexing the guides. We have a better probe. We work continuously on better probes, especially those that are better quenched. So we have a better specific activation. We improve the detection of the iPhone, and we also continuously move, on minimizing the noise. So currently we're working on our first, really design to move that prototype into an applicable point of care system. And we are working on, our emergency authorization into the next few weeks. With this, I stop. I thank the people who have contributed. It's really a relentless effort currently. Myla, Paranas, and Carly are really driving the assay side. On Dan's side, it's really Maria and Sunmin who are pushing the device side. We have many collaborators, especially also from Jennifer's lab at the IGI. BioHub gives us a lot of help with clinical samples. Google is fantastic in helping us with the, procuring the phones and also getting access to their specs. And we're also working on, putting structure around this effort currently. Thank you very much.
Thank you, Melanie. That was great. There's been a number of questions, but I think we'll try to get back on time. So if you wouldn't mind answering those questions in the Q&A box. Do you see that over there? Okay. Next up, we have Andrea Gamarnik. She is the director of the Institute of Biochemistry at the Fundación Instituto Leloir. She'll be giving a talk on COVID-19 in Argentina, from a molecular virology lab to the field.
So thank you for the introduction, and thanks to Nevan and Jacqueline for inviting me to participate in this great symposium. What I'm going to do today is to share with you what is going on in Argentina with COVID-19 and give you a snapshot of what is going on in South America. And also, I would like to share the work that we are doing in our lab. My lab is a lab that does molecular virology, basic research on dengue and Zika viruses. But recently we switched and we became a factory of to produce coronavirus proteins and to produce serologic assays to really help for this emergency. This work has been amazing. Was an amazing experience, personal experience, a group experience, and also an opportunity that we have as scientists in Argentina to give back to our society what we originally got, because in Argentina the university is free, so we study for free, and this is a great opportunity that scientists have to give back to society all the effort that we got. So I'm going to be brief today, and I'm going to start telling you that in Argentina, the first case of COVID-19 was on March 3rd, but very quickly the government took actions, and we entered in a nationwide lockdown in March 19. The government really gave a clear message to the society that what we needed is to take care of the people, and the economic situation was in a second level. So this lockdown was very strict. For instance, people that were driving in the street were stopped by the police, and if they didn't have a permit, they went, they removed the car, and people that were going out and they couldn't justify why they were walking in the streets, they had to face jail. So it was really strict, and all the universities were closed, of course, and the research institutes were shut down. But very quickly, the Ministry of Science recruited scientists all over the country to create this task force for the emergency that was called Coronavirus Unit. As I mentioned a minute ago, in Argentina, we started with 2 cases. The first case was in March 3rd, and this started growing until we enter into this very strict lockdown. And we were able, as a country, to respect this, and we very effectively flattened the curve. And we can see in the next slide that this curve was completely flat for more than 2 months. But after this time, and I'm talking about last month, circulation of people and opening of certain activities led to this sharp increase of cases. And as last night, we had almost 2,000 cases in Argentina. So in the region, we are having a lot of problems in South America. And if we see here in this plot, we can see Brazil and Chile are perhaps the worst countries in the continent. And what's going on in Brazil is really, really a big problem. There are huge, the numbers are huge, and the country doesn't have a good health care to cope with all these cases. And we can see that in one day, Brazil is having the amount of cases that Argentina had in the whole country. And the problem here is that South America is entering in the winter, so this is going to overlap with the flu season, and this is going to be a big problem. And we think that the epicenter of the pandemic is going to start switching, unfortunately, to South America. And besides that, the governments of Brazil and Chile have denied the problem since the very beginning, and even encouraged people to don't respect the quarantine. So it was really a huge lack of responsibility. So if we go back to Argentina, what we see is this is a map of the country. We can see that more than 90% of the cases are centered in Buenos Aires, in the city of Buenos Aires, and in the greatest Buenos Aires, that is the rest of the province. And inside the city, there are really hot spots that are very poor neighborhoods, very vulnerable neighborhoods, like the Villa 31 that is in this slide, where we are currently working on, that has an enormous amount of cases. So in my lab, as I mentioned before, the goal was to develop and produce a serologic kit to detect antibodies against SARS-CoV-2. We created a team of about 10 people, and we accomplished all these tasks. We generated different expression vectors to produce the coronavirus proteins. We express large amount of proteins, especially spikes, the domain, the RNA, the receptor binding domain, and several domains of the nucleocapsid. And we developed an ELISA kit. We did the optimization, validation, and so on. And we got together with the laboratory, the Lemos laboratory, that has a great experience in producing ELISA kits, because it's the laboratory that produces the kits for Chagas disease for Argentina and Latin America. So in this process, it was of great help the contribution of Florian Kramer from the Mount Sinai, who shared very generously protocols and expression vectors to start this work. Then we realized that to provide ELISA kit for the whole country, we needed to produce an enormous amount of protein. So we needed to generate new vectors that were more efficient and produce the amount of protein that we needed. And in only 1 month, we were able to come up with this prototype, this ELISA kit that was sent for external validation. It was sent to 5 different hospitals and to external quality control departments that evaluated the sensitivity and the specificity of the assay. So we tested about 3,000 samples to analyze the performance of the kit. And by May 4th, a little more than a month since we started this work, we were able to get the approval of the test by the ANMAT, that is the administration. It's like the FDA, but in Argentina. We distributed more than 30,000 cases. I'm sorry, 30,000 tests. And this distribution was free for hospitals and for health centers, private and public. So everything was given for free. The whole project took about $40,000, and now with some donations, we are able to increase the production capacity of this project of at least 50,000 tests a week, and we are going up. So in addition of becoming this factory for protein purification and ELISA kit production, we engage in a number of activities with different hospitals and institutions and with the government. We work in serology tests for hospitalized people in different hospitals. We did seroprevalence studies in different neighborhoods of Buenos Aires, And we collaborated pretty much with all the hospitals in the country that are working with convalescent plasma from donors for treatment and for clinical assays. So we were able to process and titer the IgGs in our lab, or in the cases that was possible, we distributed the kit to other hospitals that were able to, with our guidance, titer the antibodies themselves. So here we can see some of the data that we collected. We created a huge database. Here we have more than 1,000 samples from patients that most of them were hospitalized. And we can see the IgGs measurements, the signal, and the days after symptom onset. And what we learned that, I'm not going to talk about the data of this database, but what we learned from here is that the IgG appears pretty much at the same time that IgM. So at the beginning, we were going to make two kits, one to measure IgG, another to measure IgM. But based on this and all the experience that we got, we decided to use the IgM only for research or by request, and we focus our production in kits of IgG. So we increased the amount of kits that we could produce to distribute to the whole country. And here we can see that as a function of time, the IgG levels stay high. The IgM also stayed quite high, even after around 2 months. And the seroconversion was very variable. We observed a wide variety of responses. Here I show you just as an example about 20 samples, 20 patients, where we can see seroconversion very early on, few days after the onset of symptoms, or after 2 weeks or even 3 weeks. So from this experience, then we move forward with the project and focus the production in kits of IgG. And we engage in all these different projects that I mentioned. And I'm going to be very brief to tell you a few of these projects. One of them was to start this seroprevalence study in vulnerable neighborhoods of Buenos Aires. And the first problem that we ran into was that it was impossible to take samples, blood samples in the streets to the people, of course, to then take the blood to the lab to do the ELISA. So we implemented a new tool to perform the ELISA IgG assay by using finger prick blood samples. So we adapted a kit that was previously used for Chagas disease, and we validated it using a great number of blood samples and serum samples to see if this test was able to be done using blood directly taken from the finger. And the validation was beautiful. So we started this project about 2 weeks ago, a little more than 2 weeks ago, in the Villa 31. And we performed this pilot study already, and we collected participants from the streets. And also we went house by house. We are collaborating with a great team of people from the city of Buenos Aires. These people have done an amazing job here in this picture, going to the houses. These houses are really overpopulated. There are 4 or 5 families living in the same house. Or we put the tables in the street and people came voluntarily. Everybody wants to get tested. So from this, we got a seroprevalence from the pilot study of more than 10% in this particular region. And now we, with the map and with a group of epidemiologists, we have contributed to the strategy to do this in the whole neighborhood, and this is what we're doing right now. And I want to highlight the work of the epidemiologist and Alicia Mischenko, who is the person in the hospital that is doing all the clinical aspect of this. So the other work that we are doing that I mentioned briefly is to help all the hospitals in the country that are collecting convalescent plasma from donors to do this treatment or clinical trials. So we have collected an enormous amount of data. Here I'm showing you about 156 patient samples from convalescent plasma donors. And the data here are the OD, and we titer them, we titer the IgGs, and our data shows that a large proportion of these have medium 1 to 800 to large to high amount of IgGs. Of course, there is a great distribution. There are patients that have very little amount of antibodies. And there are some patients, samples, that we notice that have extremely high titers and neutralizing antibodies. And these titers are in the order of 1 to 50,000, which is very interesting for this kind of studies. And for this, we are using neutralization assays to correlate the ELISA assays using a pseudotype virus that was provided by Ben-Hur Lee, also from Mount Sinai. Ben-Hur has been also very generous providing the viral stocks, and the system is working beautifully because the readout is luciferase, so it's quite easy. So what we have done so far is to work with multi-center studies in the whole country. And of course, the question remains, what is the threshold activity or the neutralizing activity that we need for a clinical, for activity, for use in the clinic. And we won't know that until the outcome of the clinical trials come out. So with this, I would like to finish and mention the people in my lab that have done this terrific work. I mean, they are molecular virologists, and they were working on dengue, and suddenly we switch off and switch on this other project, and they have done an amazing work. Also, some investigators from the Leloir Institute, from the Lemos laboratory, and really was a great experience doing this project. And we are still working. We are working 15 hours a day and without weekends. And we are very happy to provide this tool for the whole country. Thank you.
Yeah, thank you very, very much for telling us about the situation and your work in Buenos Aires and serology testing. It's fantastic. One question by Ava Harris. Have you investigated the kinetics of the IgA response in your sample sets? You had a beautiful isotype.
It's very, very interesting, because we started doing IgA currently, and because we have a lot of samples, we can go back and look at IgA. That is something that we are very, very interested in doing.
Beautiful. Thank you. There's a number of other questions. I'll have you go answer them in the Q&A box, and we'll jump from Buenos Aires to Paris. Next up, we have Christiane Gerke from the Institut Pasteur. She's the head of the vaccines program, and she'll tell us about vaccine development for COVID-19. Thank you. Yeah. Professor at the School of Molecular and Cell Biology and Biotechnology at Tel Aviv University. And she'll tell us about full genome virus sequences and how that informs patterns of SARS-CoV-2 spread into and within Israel. Hadi.
Why could I not swap screen? Hi. Can you hear me? Yeah.
We can hear you, and the slides look great. Thank you.
Okay. So first, thanks to Nevan, and thanks to all the organizers for the opportunity to tell you about our work, about the spread of the virus in Israel. And I'm actually going to begin with my thank you slides, since it's a big slide and a lot of people to thank. This was similar to what a lot of people have been saying, a huge collaboration, a huge effort that we performed kind of during complete lockdown in Israel. So it's a really, really crazy times. The project was led by Danielle Miller and Omer Tirosh, two fantastic graduate students, and an additional 3 graduate students that you see here on the bottom accepting the samples. And we worked hand in hand with Michael Martin and Katja Cole from Emory University on the mathematical modeling end. This actually all started off similar to what Andrea described. The Israeli Ministry of Health, as the sort of epidemic began in Israel, they reached out to the Israeli academia to try and they created something which is called out of the box work group, to try and think how academia can really help the effort in fighting the virus in Israel. And this really led to many thoughts about how we can actually aid using a lot of our knowledge in how to sequence the virus and how to analyze data from sequencing to really gain a better understanding of the epidemiology of virus spread. And our initial questions, which we kind of eventually reached kind of different sort of avenues with this project, but initially we were interested in trying to estimate the incidence of the virus, the total number of infected people in Israel, which includes the asymptomatics. And this is something which is still today sort of this big open question. How many asymptomatics are there, and how many people are the tests missing? And the second question was to estimate the basic reproduction number of the virus, so-called R0. And the idea is that really the traditional epidemiology is questionnaire-based. It's often very slow, and the biggest problem is that it could be biased. And whereas when we have sequencing, I mean, sequences don't lie. And the idea is that we could use sequencing to try and answer some of these questions as quickly as possible. And so what do we know so far about SARS coronavirus 2 in terms of the evolution of the virus? We know that there's clock-like evolution of the virus. Mutations accumulate at a set pace of 2 mutations every month, which is more or less equivalent to every second transmission. A mutation is fixed in the virus genome. We know that the absolutely vast majority of mutations are neutral, which means that they do not affect the phenotype of the virus. They don't have any effect on any function of the virus. And so in this kind of toy illustration here, right, we have a person that is infected. If this person will infect someone else, on average, the genome will not change, but the second time a person is infected, there will be some random mutation that will fix somewhere in the viral genome. And this, of course, repeats over and over. And I gave this toy illustration because what we do is we actually use these mutations as barcodes that allow us to track viral spread. And so the first stage of this work was that we collected 212 samples. Actually, originally we collected a larger number of samples, and then we weeded some out, as I'll soon describe. These are sort of leftover RNA from the swabs that I guess everyone is familiar with by now, the same swabs that are used to detect viral presence. And our criteria were, and these are important criteria, is that we assembled samples from different dates spanning from March into April. Our last sample is dated April 22nd. That's when we started analysis. We excluded any relatives, and we sampled from 6 hospitals spanning the entire geography of the state of Israel. And why are these criteria important? Because basically our aim was to gain a random representative sample from everyone that was infected in Israel. The second stage is that we performed full genome sequencing of the virus from these 212 samples using next generation sequencing. The genome length is about 29,500 bases. And we really did succeed in getting high coverage and high quality sequences from all of the samples. And so what did we learn from the sequences? And so for the first thing I'm showing you here is a map of the mutations that we see along the genome. The x-axis is the location in the genome. The y-axis is a count of number of samples with a specific mutation. And what I think you can see clearly here are that mutations are spread all over the genome. We actually have an equal number of, twice the number of non-synonymous versus synonymous mutations. And this is kind of an indication that there is no selection operating on, at this stage, there's no selection operating on mutations. And that was important for us because selection would skew the signal that we looked at later. 8 sequences harbor deletions, so-called microdeletions, very, very short deletions. The shortest deletion was 1 nucleotide, the longest was 18 nucleotides. We were actually quite surprised by these deletions. 3 of these 5 deletions are in-frame deletions, and they're less surprising because they don't change the open reading frame. But 2 of them are not in frame and affect ORF7A and ORF8. And in fact, the deletion that affects ORF7A actually basically wipes out this gene. And this was very, very surprising. But then we went and looked into the emerging literature and found out that this has actually been reported also. So in Singapore and in Arizona, deletions, larger deletions have been affected that affect both these genes. A very curious thing that we noted is that 4 of the deleted samples reside in the same clade. So this is the tree, the phylogenetic tree of the Israeli sequences, and 4 of the deleted samples reside in the same clade. We're not sure of the meaning of this yet, but this clade is actually defined by one non-synonymous mutation at NSP16. And at this stage, all we can say is that it seems that SARS coronavirus 2 is prone to these microdeletions. Others have reported this as well. And it seems that the virus can tolerate these deletions to some extent across different regions of the genome. The next thing we went on to look at is to compare the Israeli diversity to the global diversity. And here we use this wonderful resource called NextStrain, which I'm sure some of you are familiar with. And so what we see here is the phylogeny of sequences. Today we already have something like 50,000 sequences that have been sequenced, and this is a subsample, a representative subsample of 5,000 sequences. In purple we see sequences from China, and greenish we see European sequences, and red are sequences from the United States, and the blue dots are the Israeli sequences. And what we can actually see here quite visually is that most of the blue dots reside within sort of red regions, which are the United States. And when we went on to quantify this by looking at the map, we saw that 70% of transmission chains in Israel are derived from the United States. The rest are more or less mostly from Europe. We see almost no transmission chains in Israel from Southeast Asia. And this was really surprising. We were so surprised that we went on to check other countries. This is not a pattern that we see anywhere else in the world. And we went on to compare this to data that we have from the Ministry of Health. So the Israeli Ministry of Health has been really closely monitoring any returning travelers and whether they are infected. And we looked at the proportions here, and we saw that only 27% of infected returning travelers were from North America, and this is almost exclusively the United States. Whereas our sequencing data suggests that 70% of what we call clade importations or transmission chains in Israel are from the United States. And so there's a strong discrepancy here. And what we actually infer from this discrepancy is that travelers returning from the United States contributed dramatically to spread in Israel. And we went back and looked at the dates, and actually this was a big issue sort of politically in Israel. So travelers that returned from Europe were put into quarantine as of February 26th. Whereas from the United States, they were not. So there was a gap in policy that lasted about 2 weeks, where during these 2 weeks, what we think happened is that people returned from the United States. They didn't enter quarantine because, of course, they weren't supposed to. And this led to the fact that they spread the virus in Israel, and that's why we see this such a large proportion of transmission claims from the United States. The second thing we went on to do is, this is again working with Katya and Mike at Emory, as we fit an epidemiology model called a susceptible, exposed, infected, recovered model. And the most important thing that this model takes into account is first of all, that some people might spread the virus more than others, and this is called transmission heterogeneity, and I'll get to this in a minute. Second of all, we assume that there's a non-infectious curious following exposure, and this is important because we know that there's actually a very unique sort of dynamic of the virus, of this particular virus. And we actually directly model the possibility of transmission dynamics changing following social distancing measures. And so we know that there's R naught that is associated with viral spread when there's no social distancing measures, and we wanted to test how social distancing, if it affects R naught. And so first of all, looking at this R0, so R0, just to remind you, is the mean number of individuals that every person infects. And so we see that before March 19th, and so March 19th is when there was a national lockdown, a statewide lockdown in Israel. So the strongest, basically, social distancing efforts that were implemented in Israel. Before March 19th, R0 was around 2.3. And after March 19th, and this is again only based on the sequencing data, we see that there's a very strong drop in R0, going down to 0.7. We actually know that later on it probably went down even more than that. And unfortunately now in Israel, there's something which is similar to a second wave. And there are actually, there's an increase in R0 today and more and more cases that are detected. This is really, I think, very unfortunate. And so this is what we see again based on sequencing data only. And now going on to transmission heterogeneity, we know that R0 represents an average. Okay, so it represents an average across a large number of people. And in the field of infectious disease, there's something which is known as the 20/80 rule. And so for many infectious diseases, we know that 20% of infected individuals are responsible for 80% of cases. So this is kind of illustrated here. And these are so-called super spreaders. And so we actually introduced this into the model in our earliest efforts and to test if this is also what is going on in the case of SARS coronavirus 2. And actually what we found is that this does not match what we see in our data. And so the dashed line here is the number of reported cases on April 22. And this is our inferences of the model under different levels of super spreading. So we have a parameter in the model called pH, which is the proportion of high spread patients that are responsible for 80% of infections. And so 20% is the value that is, again, known for most infectious disease. And if we assume this 20%, the model infers that there were less people infected, less cumulative incidence than the actual number of reported cases, which of course completely does not make sense. And the only values that do seem to make sense are somewhere between 2% and 10%. So if pH is between 5 and 10%, it seems that we more or less hit the number of reported cases. And if 2% are responsible for 80% of infections, this means that there's underreporting. But all in all, what we see here is what we call extreme super spreading dynamics. So 2 to 10% of patients are responsible for 80% of infections. And with that, I'll summarize and I'll end, and I'll say that so what we saw here is that transmission from US travelers was very, very dominant in Israel. We think that this is a gap in policy that was because European travelers were quarantined early on and United States were not. We see this very strong effect of super spreaders. We don't know if this is biological, so associated perhaps with some people for some reason, perhaps for bearing more virus, or for some other reason, some biological reason, able to spread the virus more efficiently, or whether this is something that is social and has to do with large gathering. And so we know that there's more and more evidence and more reports on the role of large gatherings and how they affect spread. In Israel, actually very, very recently, one high school was associated with 150 cases probably derived from one or very few people that were responsible for infections. And this is something that is seen around the world. As a corollary, what we actually learned from this is that it is possible that a large number of individuals actually do not contribute to spread at all, or contribute very little to spread. And finally, we see that the sequencing data alone shows the effectiveness of the shelter-in-home measures that were implemented in March. And with that, I'll end. Happy to take questions.
Hadi, thank you very much. That was fantastic. Thanks for giving us the insight into how the quarantine rules influenced the coming into Israel with the cases from the US. That was fascinating. One question is from whether the genome sequences are made available through an open repository?
Yes, they are. So we immediately uploaded all our sequences into GISAID, so the public repository that harbors most of the sequences for SARS coronaviruses, and they're labeled as Israeli sequences, and they're also available on NextStrain. So NextStrain subsamples some of the sequences, and they're available there.
And then another question about what is the reference sequence? I think this is, how do you find if any mutation occurred? Is there a patient 0, or do you just compare them with time?
Yes, so the reference sequence is the first sequence that was sequenced from Wuhan in China. And that's used in all typical studies as the reference sequence to compare against.
And we find maybe that there might be an earlier version, will that reference be readjusted, or?
That's a good question. Well, it should be. In theory, yes, we should be adjusting it. I think I'm not sure. I'm not sure about the meaning of it, because I mean, what it would do, it would shift like a whole lot of, maybe like 2 or 3 mutations would be shifted into becoming mutations versus non-mutations.
Right.
I don't think it would have much of effect on our analyses in general.
Great, thank you, and thank you for staying up so late to participate.
Yeah, thank you.
Now we're going to be coming back west to New York. We have Viviana Simone, Professor of Microbiology, Medicine, and Infectious Disease at the Icahn School of Medicine at Mount Sinai. And the screen looks good. And Viviana will tell us about Mount Sinai Goes Viral, SARS-CoV-2 surveillance.
Let's see.
I think perfect.
Excellent. Thank you, guys. I'm going to this was a great introduction and follow up to Adi's talk. So we are going to talk about viral surveillance here in New York City. Okay. Let's see. Not sure how to forward here.
Sometimes just use your mouse, maybe. Try to.
Here we go. So New York City is really a very international hub. We have lots of visitors from inside the United States as well as from all over the world, which makes it a great place to visit and live. But also we are very, very diverse with respect to our pathogens. So we think that New York City could very likely be an entry point for pathogens of pandemic potential. And we worried about this quite a while, because Mount Sinai is a large hospital system here in the metropolitan area. We have 8 hospitals that are part of this health system. We serve a large, diverse patient population, and we also have a large workforce of approximately 40,000 employees. We see a lot of respiratory infections here in the city. On the bottom, you see the influenza numbers over the past 3 years. So we were worried that if there would be a respiratory virus that would spread through the world, New York City would be especially vulnerable. In order to address this, we built the Mount Sinai pathogen surveillance program. It was initially intended to follow pathogens of bacterial origin and fungi that were found in our patients in the hospital system, especially in the context of nosocomial infections. In 2017, we started expanding the surveillance program to respiratory viruses. As of now, we have banked over 13,000 specimens from patients with viral infections. Most of them are respiratory infections, but we also have a couple of herpes viruses, CNS viruses, and such. Over the past 3 months, we have banked over 3,000 nasopharyngeal swabs of SARS-CoV-2 infected patients receiving care at Mount Sinai, as well as plasma and serum. Importantly, all those samples are banked with a linking key so that we can access demographics, clinical outcome, vaccines, pre-existing conditions, and outcomes of the infection. And we have excellent sequencing facilities and pipelines that allows us to use both PacBio and Illumina for sequencing the pathogen genome. Like many others have said, this is a large team effort. So this pathogen surveillance program is led by Harm von Backel here in the center, and he's a professor in the genomics department. The other important stakeholder in this enterprise is the Dr. Sodillo from the clinical pathology lab, who leads the microbiology clinical microbiology. Dina Altman is an faculty in the infectious diseases. So work I'm going to, on the left side, you see the COVID task force of the Simon Lab. And so the data I'm going to present is driven and led by Ana Gonzalez Reiche here on the right. So in before COVID times, our pathogen surveillance program focused on following seasonal influenza viruses. We looked at the frequency in the hospital system. We tracked on a routine basis the locations of the viruses. We sequenced their genomes and also actively monitored and tracked nosocomial outbreaks in our hospital system. On the right side, you see that we use the genetic information for surveillance. And from that information, we knew that New York gets a lot of pathogen from Europe. So we often, with influenza, we reflect more closely the subtypes distribution seen in Europe compared to the other part of the United States. But we also noted that New York City has the pathogens that cause disease in our patients represent very well the global diversity of disease-causing influenza viruses. So with this, we basically had an infrastructure in place, and when we heard about this new virus emerging in China, we basically retooled this infrastructure to study coronaviruses and should it come to New York. And that happened on February 29. The first case of corona was identified in a person seeking care at Mount Sinai. Both the first two cases seen at Mount Sinai actually were individuals with a known travel history. So New York 1 had a travel history to the Middle East, and New York 2 had a travel history to Europe. In both instances, those infections did not result into forward transmissions. So they were limited to those individual introductions into New York City. We then embarked into sequencing. Oh yeah, this shows you, this is a quite dramatic slide. Just shows you the number of patients that we had over the past 3 months, starting in November with influenza A and B. And this was quite a severe influenza season here in New York City. But compared to what we encountered with SARS-CoV-2 in New York City, it really is dwarfed. So here, New York 1 is the first case identified end of February, and you can see within 2 weeks the SARS-CoV-2 infection rate exploded, reaching a peak early April. And then fortunately, all the containment measurements kicked in, and we could control and stabilize the infection rates. The data I'm going to show you is going to be just the beginning, just the upkick of the infections. We sequenced 84 viruses from 84 patients in this early period of time. And this is a phylogenetic tree shown on a time scale at the bottom. And we depicted here the regions from which the viruses came. And you can see that there are 10 different introductions that we could reconstruct in this phylogenetic tree. And 5 and 6 are New York 1 and New York 2. There's the traveler-associated introductions. And here we see a cluster that suggests community transmission resulting from untracked transmission. We looked further into those as evidence for community transmission, and there was the first cluster of community transmission in New York City happened in Westchester, and you can see that here we have one strain that originates from Westchester County, and it closely clusters with a number of viral isolates that come from patients living in the Bronx, King County, Queens, Westchester. Those are the boroughs surrounding New York City or in New York City. So there is clear evidence for local transmission clusters in our data set. And we identified the signature mutations for those clusters. The next question we wanted to answer is, can we use this sequence information to basically track back when a possible introduction occurred? So for that, we looked at the most recent common ancestor in this phylogenetic trees. Here again, we have the two cases with an epilink to travel, but we have a number of cases in which we can compute that the introduction probably happened early February to late January, with a large range of uncertainty for some cases. So as the pandemic continued in New York City, we expanded our sequencing efforts to now way over 300 complete genomes. This is the same representation Adi showed. It's generated on next strain. All the red dots are individual infected patients seeking care at Mount Sinai. And you can see, not unlike with flu, we have basically viruses that represent the global diversity of viruses circulating. Here we have, this is basically up to the peak of the pandemic in New York City. And then we have sequenced the next round early May. And you can see that this is basically here the continuation of what was the beginning of the community spread here. So the dominant strains here are of European origin or intermingled with European viruses. There is not enough diversity to really have a directionality in this data set, but we can say that the viruses in New York City resemble the viruses seen in Europe. A lot of attention has been given to this mutation in spike at position 614, which might be associated with more rapid spread. So this is the same tree I just showed you. And now I have colored the viruses derived from the patients at Mount Sinai, depending on whether or not they have the wild type or the mutation. And you can see that the tree really falls into two parts. In the beginning, we had co-circulation of strains with 614D and G here on the bottom, whereas at the later time point we see that it's mostly the G variant that is dominating. We are sequencing more viruses to make sure that this is not only due to a lower sampling. Since we have linked information to the disease outcome, we were interested if there's any evidence to the fact that some viruses are more pathogenic than others. Same representation as the previous slides, only now that we colored the viruses from patients depending on the disease outcome. In blue, you see the patients that were discharged. In yellow are the patients that are still in the hospital. And then in green, you have the patients that passed away. And you can see the yellow dots are actually distributed throughout the tree here without any clear evidence that one strain is more deadly than the other. It seems if you normalize it by the number of cases we have, it's quite equally distributed. But we are following up on this. This is not yet a complete data set, so we are going to try to fill in more information regarding the disease outcomes for the individual patients for which we have sequences. So the next thing is, of course, we are interested how things change over time. Here, this is a deconstruction of the frequency of SARS-CoV-2 clades over the past 2 months, with a focus on the US coast. And here, mentioned earlier, Wuhan 1 is the virus that started. It's used as a reference here. And you can see how the frequency changes quite dramatically. And we have a couple of viral clades here in New York City that are quite dominant, but change over time rather quickly. So this will be very interesting. We will use this information together with the information from the previous slides to generate a reference set of viruses that recapitulates global diversity, as well as what we see here in New York City. And we are going to do this in a systematic manner so that we can assess the phenotype of those viruses and provide those viruses as resources to the scientific community. So to conclude, SARS-CoV-2 has had immense impact here on New York City. It has been one of the early epicenters of the pandemic in the United States. We have over 200,000 confirmed cases in New York City. We showed that this outbreak in New York City was a result of multiple independent introductions with local forward transmissions due to undetected infections. As mentioned, we are going to use this sequencing information to generate a reference set of viruses to characterize in cell culture and in animal models to really understand the phenotype of the disease-causing viruses. We will also continue with our ongoing surveillance, and we want to see if we can pinpoint the impact of public health measures on the community transmissions. We are also looking into how to map the interaction between SARS-CoV-2 and the seasonal influenza viruses coming fall. So to see how those two respiratory viruses shape the pathogenicity of each other. And last but not least, we also are quite concerned about nosocomial transmissions in this context, and we are looking to contact tracing if those instances should become apparent in our hospital system. So this, with the acknowledgements, it takes a village to get this off the ground. Again, this is a collaboration, ongoing collaboration between the Van Bakel labs, the Simon labs, the clinical microbiology lab, and my colleagues in the Department of Microbiology and Genomics, Genetics, as well as my clinical colleagues in infectious diseases, infection prevention. And the sequencing core also gets a shout out. Thank you all for your attention, and I'm ready to take questions.
Thank you, Viviana. That was great. Thank you for telling us about how you persevered during this onslaught on New York, and were able to, that view of the sequencing clades with time is beautiful. One quick question, do you think environment can affect the mutation rate in patients? And then we'll have the rest of the questions in the box if you could answer.
I certainly New York City, because we have such a dense patient, we live in so close quarters, we take the subway. I could imagine that, this was really facilitating the spread of the virus, and with now shelter in place orders, that clearly has had a positive impact on curbing the transmission.
Great. Well, thank you. Stay well. Okay, Nevin, you'll take over from now.
Yep, I will jump back in to introduce our last speaker, the present chair, Kevin Shrokat. He's a professor in the Department of Cellular and Molecular Pharmacology at UCSF, and also an investigator of the Howard Hughes Medical Institute. And one thing that I've learned actually through this pandemic is Kayvon's actually available 24/7 when you text him or call him. So that's pretty impressive. I don't know if that's going to stay in place, Kayvon, when the pandemic goes away, but it's been great to work with you. And obviously you've been a key part of the QCRG, and you're going to be discussing today leveraging new insights from cancer drug discovery to target viral host factors.
I'll do my best to always reply in a timely manner. Thanks, Nevin. I'd like to tell you about efforts from the drug discovery subgroup that Brian Shrokat and I head, and a particular project that we've been working on with another subgroup leader, Davide Ruggiero, that is running the translation group. And this has all been done in collaboration with the entire QCRG, but very much informed by the bioinformatics and systems biology groups run by Pedro and Nevin. So this sort of fire hose of data since March has been amazing, and we've gotten the benefit of two fantastic data sets. The one that Nevin spoke about, the protein-protein interaction map, developed by the Gordon, and reported in the Gordon paper. And then Danielle mentioned the newly obtained phosphoproteome and proteome data that is very, very complementary, because this is monitoring phosphorylation and protein abundance status in SARS-CoV-2 infected viro cells. And so by looking in these two data sets, Brian and my groups, as well as all the other groups in drug discovery, have been trying to mine many of the nodes here and try to place drugs that might interrupt the viral factors that are co-opting host factors to carry out the life cycle and replicate. And I'll tell you about one such effort. So, so we know the SARS-CoV-2 virus is an RNA plus stranded virus. So when it enters the cell, it has to execute translation of its mRNA to produce protein and start reassembling the next virion. And this process is very, very highly regulated. The proteome data now that we have on the right is really fascinating, because you'll see that in the blue dots, these are proteins that are downregulated at the 24-hour time point, and in red are the proteins that are upregulated. In the open red circles are the viral proteins, and in the closed red circles are the host proteins. And you see that by and large, all of the viral proteins are massively upregulated, but the host cell proteins are really not upregulated. And in fact, many of the host proteins are downregulated. So this is an amazing machine that the virus can come in and co-opt our translational machinery to translate its own mRNAs and suppress its own mRNAs and suppress the host RNAs. So how does it do that? Now, we, many of us in the QCRG are really in our daytime jobs, we work on cancer. And so we tried to think about how cancer cells co-opt translational machinery in order to produce proteins from onco-mRNAs. These are so-called onco-mRNAs because translation of them are correlated with oncogenesis. And if we look at the onco-mRNAs such as cyclin D and MYC, these are highly upregulated when the growth factor pathway is turned on. This can be turned on in cancer cells by mutation or the presence of excess growth factor. And when all of these factors are up, you see at the bottom of the cascade, cap-dependent translation of cyclin D and MYC is very high. So we took the early simple hypothesis and asked, well, is the SARS-CoV-2 infection somehow like the is the virus reprogramming normal cells somewhat like a cancer cell reprograms a normal cell? And if so, can we repurpose cancer drugs to treat SARS-CoV-2? So we had a number of drugs, and I'm not showing many of them that we tested, but two of, I think, the most selective molecules to block growth factor signaling are rapamycin and sapanisertib, another mTOR inhibitor. And you see these are about midway down the growth factor pathway, but right before the translational machinery is turned on. And what you'll see on the right, data from Chris White and Adolfo Garcia-Sastre from Mount Sinai, is that the rapamycin and the sapanisertib drugs were basically in red are the percent infection rates at different doses, and they have really no effect on viral replication. Whereas, as Adolfo mentioned before, these inhibitors suppress the host cell from replicating, but they don't kill the cell. It plateaus at about 50%. But this is not what we want. We want something that has anti-viral replication activity, but these obviously don't. So we had to readdress our hypothesis and think what else is going on, because obviously the upstream instructors of the translational machinery are not turned up in viral replication. In fact, that's also emerging from the phosphoproteome data. But what it told us is if the viral proteins are getting translated, this part at the bottom of the cascade, the machinery, if you will, is turned up. And so we were very, very fortunate to have Davide Ruggiero here at UCSF. He's one of the world's leading experts on translational control in many settings. And so with him, we began to think about what parts of the machine might the virus be co-opting. So let me give you a quick introduction to how mRNAs are translated in a selective manner. All these RNAs are cap dependent, so they have a 7-methyl G at the 5' end. EIF4E is a factor that recognizes the cap. It recruits another scaffold protein, EIF4G, which has the ability to recruit other factors as needed to regulate the 5' untranslated region and recruit ribosomes for translation. So in many important RNAs, there are 5' structured RNA elements that block the scanning by the 40S ribosome. These mRNAs require other factors. The one I'll really focus on here is the RNA helicase EIF4A, and it has its own factors like EIF4H that activate its activity. The helicase comes into the 4G complex, scans along, finds a structured element in ways we don't fully understand, and melts the structured element, allowing 40S to scan, and now it bypasses the structured element, finds the start site, and then assembles the ADS. So now with that thinking in mind, Davide looked into the 300 or so protein-protein interactions between viral factors and host factors, and noticed that NSP9 had one node of interaction with EIF4H, which, as you'll notice, I mentioned before, is a specific activator of EIF4A, the RNA helicase. That led to the hypothesis that the virus relied on 4A to unwind structured RNAs in its own 5' UTR. Simultaneous with this insight, groups at Stanford, Maria Barna and Riju Das, Maria's lab works on translational control, is expert there. Riju is an expert on RNA structure. They began to look and analyze the 5' UTR of SARS-CoV-2, and it looked like there might be sequences that 4A could unwind. So with that notion in mind, we started to see if we could target EIF4A, this host factor, and its ability to unwind the 5' UTR of SARS-CoV-2. Here is where natural products and evolution really served us incredibly well, because nature already came up with what I think could be a perfect drug for this host factor. So as 4A is scanning along for structured regions, it then will unwind. It's an ATP-dependent enzyme, but this natural product, a rocoglamid, also known as a sylvestrol molecule, is a molecular glue that can trap the helicase with the RNA and the protein interface. Now, this is a very unusual way for drugs to work. Nature finds this many times, but as chemists, we often don't design them this way. But this has the chance to be, I think, a fantastic drug, because although, as Nevin said, we were targeting host factors, this drug lets us use the host factor 4A and maybe trap it on the viral RNA. So in a sense, it's a direct acting drug because it might directly act on the viral RNA. This is something that Davide's lab, Joanna and Tuigu are working very hard on by studying to find what sequences in this 5' UTR might be sensitive to 4A. But this is a mode of action that has only recently been discovered with this beautiful crystal structure from the macaque et al. Groups. And here you see in cyan, the rocoglamid sits with the RNA and traps the RNA that the RNA helicase is trying to unwind. And the great thing about this is it acts in a dominant way, because if this drug can trap helicase, then no other helicase can come on and unwind that. Now, we were also fortunate that although natural products sometimes can directly become drugs, these rocoglamids themselves were not suitable because they didn't have optimized drug properties. I should also note, when we were starting to look into this, we found evidence that other groups in just a few years ago had shown these same rocoglamets had antiviral activity in coronaviruses. So that became exciting. Then we also benefited from Effector Therapeutics, a company that Davide and I co-founded with Steve Warland, who had turned the natural product into a suitable drug candidate that it had just put into clinical trials a year before. And Chris and Adolfo's lab was able to obtain data to show that zotatifin has really potent antiviral activity. Its most potent, its activity is 37 nanomolar at blocking production of viral particles at its IC90, which is probably one of the most important sort of metrics of how a drug can block viral replication and reinfection. So Effector is working hard to take this drug forward and file an IND, change the IND from the cancer indication to the to a COVID-19 population. So we're hoping that that would come through. Very excited to hear about that and developments there. So with that, I just want to again thank the fantastic drug discovery group that Brian and I helped co-lead, but it's really a very fantastic group of faculty, students, and postdocs who've all worked together to mine this fantastic data set. And the translation group, especially Davide and Stephen, have been instrumental in helping us think about mechanistic cell biology in light of the viral infection. I'd like to thank the entire QCRG, especially Adolfo's group, and DARPA and HHMI for funding, and Steve Warland, CEO at Effector, for working with us on this. Thank you.
All right, fantastic, Yvonne. Very good. Very exciting. Got a few questions. What is the implication if we trap EIF and polypurine RNA sequences for a long time? I guess the guess is this would be an acute therapy?
Yeah, great question. Yeah, I think that we already know that patients in the cancer trial have been dosed for several cycles, so months. And so we think that that is tolerable. Very interestingly, the drug has a biodistribution profile that's very favorable for COVID-19 patients. It concentrates in the lungs. So we think that, as you said, this would probably only be a 1 week or 2 week, just like we heard about the remdesivir trial. So that's very, very likely to be tolerated, but we know that this drug is even tolerated for longer time. So we have a good indication already of its tolerability.
Another question here. Can roclamide interfere with EIF4A, the helicase of exon junction complex affecting mRNA decay or splicing? I guess what's the mechanism there?
Yeah, I think that's a great question. I don't know whether we know all of the RNAs that helicases really work on. That's a challenge that we've been working on in my lab with other helicases. But I think that because we have the rocoglamid, we could figure that out. It's a great question. That's where I think Davide would have to step in and help me out.
Is there a possibility of iris in SARS-CoV-2?
There is, and so these internal ribosome entry sequences sort of bypass the cap, but as I'm learning, the irises come in many, many flavors, so it's very hard to even identify them by sequence, very functionally dependent, but they're very context dependent, so the RNA world is very, very still very, very complicated. I think it's a great area that I know Davide has mentioned to me he's working on as well.
One last question I think is a good one. Are you looking to get a virus resistant to zotatifin, and would that be mutations in the interacting NSP protein or?
Yeah, we haven't done that. That's a great thing we should try to do. I think that can give us a heads up about true mechanism of action. Great point.
Okay, and there's a few other questions there I'll let you answer on the chat. But so I'm just going to share my screen just for a couple of wrap-up comments. So I just want to thank everybody, all the speakers and all the sponsors. You can see it was a truly a worldwide effort here. If you even just look at the last session that Kevon was chairing, you know, he went from San Francisco to Buenos Aires, to Paris, to Tel Aviv, to New York, to San Francisco. I think that's pretty remarkable. And if you look at the swath of what was talked about, the broad swath related to COVID-19 research in terms of therapeutics and mechanism and virology and surveillance, epidemiology, diagnostics, vaccine development, imaging, to me, it was incredibly impressive. And as scientists now, as we try to deal with, you know, interacting in creative ways, I thought it's quite remarkable how you can go around the world multiple times in a symposium like this. And to the participants we averaged about 400 or 500 participants the entire time, which I also thought was incredibly impressive. So thanks to all the speakers. Thanks to the sponsors, again, Brooker, Synthico. Thanks for the great flash talk from Synthico at the break, Thermo Fisher Scientific, as well as the host pathogen mapping initiative. And then again, just the last slide, which is probably the most important one, is thanks to these individuals that are behind the scenes, making sure everything runs smoothly. Jacqueline, Gina, Alexa, and Carolina. Thank you so very much. And again, thanks to everybody chiming in, and we hope to interact with all of you very soon. Thank you.